Hello, and welcome. It's good to have you here in this quiet moment, wherever you are. Perhaps you've had a long day, or perhaps you're simply ready to let your thoughts slow down, to let the world outside soften and fade for a little while. That's perfectly fine. You don't need to do anything right now except listen. And even listening can be gentle, effortless, like letting warm water run over your hands. Tonight, we're going to explore something extraordinary, something that's happening inside you right now, even as you rest. We're going to wander together through the calm, beautiful world of oxygen, transport, and blood. It's a story written in molecules and currents, in the silent cooperation of billions of tiny travelers, all working in harmony to keep you alive, to keep you breathing, to keep you here. And the wonderful thing is, you don't have to do anything to make it happen. It's already unfolding, quietly, perfectly, without effort. If you'd like to stay with me for more of these gentle journeys into science, you're welcome to follow along. But there's no pressure, no urgency. Just rest. Just be here. So let's begin softly at the very beginning. Every breath you take brings the world inside you. The air around you, invisible and weightless, slips past your lips, glides down your throat, and enters the branching pathways of your lungs. And there, in the smallest, most delicate chambers, something remarkable happens. Oxygen, that essential element born in the hearts of ancient stars, meets your blood. It doesn't rush or force its way in. It simply dissolves, crosses a membrane thinner than tissue paper, and begins a journey through your body that will sustain every cell, every thought, every heartbeat. This is not a violent process. It is one of the most peaceful exchanges in all of nature. Your blood is not just a red river. It is a living tissue, a fluid organ that flows and adapts, carrying not only oxygen, but also nutrients, hormones, heat, and messages. It is ancient, this fluid. The very idea of blood, of a liquid that moved through a body to deliver life, emerged hundreds of millions of years ago, long before there were lungs, long before there were hearts as we know them. The first creatures to develop something like blood were simple, soft, and slow. They lived in oceans that were quieter than ours, and their bodies learned, gradually, how to carry oxygen from the water around them to the cells within. We are their descendants, and we carry that legacy in every drop. The color of blood, that deep red, comes from a single molecule. It is called hemoglobin, and it is one of the most elegant structures in all of biology. Hemoglobin is a protein, a carefully folded chain of amino acids, and at its heart lies iron. Iron, the same element that rusts in rain, that forms the core of our planet, that was forged in the collapse of dying stars. In your blood, iron does not corrode or decay. It binds to oxygen. It holds it gently, like a hand cradling a flame, and then releases it exactly where it's needed. This is not random. This is precision, written into the shape of the molecule itself. Each hemoglobin molecule is made of 4 subunits, for smaller proteins that fit together like puzzle pieces. Each subunit holds 1 heme group, and each heme group holds 1 atom of iron. So 1 hemoglobin can carry 4 molecules of oxygen. For tiny passengers, held in place by forces that are both strong and yielding. When oxygen binds to hemoglobin, the protein changes shape just slightly. It relaxes, opens, becomes more receptive to the next oxygen molecule. This is called cooperativity, and it means that the more oxygen hemoglobin holds, the easier it becomes to hold more. It is a molecule that learns, in a sense, from its own experience. Red blood cells are the vessels that carry hemoglobin through your body. They are small, so small that 50 of them could fit across the width of a single human hair. And they are shaped like discs, slightly indented on both sides, like soft cushions pressed in the middle. This shape is not arbitrary. It gives them flexibility, allowing them to squeeze through the narrowest capillaries, the tiniest blood vessels, where the walls are only 1 cell thick. A red blood cell can fold, bend, twist, and still spring back to its original form. It is resilient, built for a life of endless travel. Inside each red blood cell, there are about 270 million hemoglobin molecules. Let that number rest in your mind for a moment. 270 million, all packed into a single cell, all working in unison. And you have trillions of red blood cells. Trillions. The scale is almost beyond imagining, and yet it is real, and it is yours. Right now, this vast fleet is moving through you, carrying oxygen to your brain, your heart, your fingertips, your toes. You are a universe of cooperation. Red blood cells live for about 4 months. They are born in the soft, spongy marrow inside your bones, especially the bones of your spine, your ribs, your pelvis. The marrow is a hidden factory, warm and dark, producing millions of new cells every second. When a red blood cell is first made, it has a nucleus like most cells, but before it enters the bloodstream, it ejects that nucleus. It gives up its ability to reproduce, to repair itself, to make new proteins. It becomes a simple, dedicated carrier, a vessel with one purpose: to transport oxygen. This sacrifice makes room. Without a nucleus, there is more space inside for hemoglobin, more capacity to carry life. When oxygen enters your lungs, it is surrounded by air, by nitrogen and carbon dioxide and trace gases. But only oxygen is small enough, reactive enough, to cross the barrier between air and blood. The walls of the alveoli, the tiny air sacs in your lungs, are incredibly thin. They are made of a single layer of cells, and just beyond them is a network of capillaries, blood vessels so narrow that red blood cells must pass through in single file. Here, oxygen dissolves into the fluid lining the alveoli, then slips through the cell membrane, through the capillary wall, and into the blood. It happens in less than a second. It happens with every breath. Once in the blood, most oxygen does not float freely. Free oxygen would dissolve poorly in the watery plasma that makes up most of your blood. Instead, it binds to hemoglobin. The iron atom at the center of each heme group has 6 bonding sites. 4 occupied by the surrounding protein structure. 1 is occupied by a part of the heme itself. The 6th site is open, waiting. When oxygen arrives, it attaches to that 6th site, forming a loose, reversible bond. This bond is strong enough to hold oxygen during its journey, but weak enough to release it when the time comes. It is a bond of perfect balance. The binding of oxygen to hemoglobin is influenced by many factors, and this is where the system becomes truly intelligent. In your lungs, where oxygen is abundant, hemoglobin binds it eagerly. But in your tissues, where oxygen is scarce and needed, hemoglobin releases it. How does hemoglobin know where it is? It doesn't think, of course, but it responds to its environment. It senses the chemical conditions around it, and those conditions tell it what to do. One of the most important signals is pH, the measure of acidity. When your cells are active, they produce carbon dioxide as a waste product. Carbon dioxide dissolves in blood and forms carbonic acid, which lowers the pH, making the blood slightly more acidic. Hemoglobin is sensitive to this change. In a more acidic environment, it holds oxygen less tightly. It lets go. This is called the Bohr effect, named after Christian Bohr, a Danish physiologist who discovered it over a century ago. It is a beautiful feedback loop. The more active your tissues are, the more carbon dioxide they produce, the more acidic the environment becomes, and the more oxygen is released exactly where it's needed. Temperature also plays a role. Warmer tissues that are working hard and generating heat, cause hemoglobin to release oxygen more readily. This makes sense. A muscle that is contracting, a brain that is thinking, these are warm, active places that need fuel. Hemoglobin responds to warmth by loosening its grip on oxygen, by giving more freely. It is as if the molecule itself understands the needs of the body. There is another molecule that helps regulate oxygen release, and it is called 2,3-bisphosphoglycerate, or BPG for short. BPG is produced by red blood cells themselves, and it binds to hemoglobin in a way that makes it harder for hemoglobin to hold onto oxygen. This might sound counterproductive, but it's not. BPG ensures that hemoglobin doesn't hold oxygen too tightly, that it remains willing to release it in the tissues. Without BPG, hemoglobin would be too greedy, too reluctant to let go, and your cells would starve even as your blood remained rich with oxygen. BPG is a mediator, a gentle reminder that holding on is not always the answer. The amount of BPG in your blood can change. If you travel to a high altitude, where the air is thin and oxygen is scarce, your body responds by making more BPG. This shifts the balance, making it easier for hemoglobin to release oxygen even when less is available. It is one of the ways your body adapts, quietly and without your conscious effort, to new environments. People who live their entire lives at high altitudes have more BPG than those who live at sea level. Their blood has learned to give more generously. Carbon dioxide, the waste product of cellular respiration, must also be transported by the blood, but in the opposite direction. It travels from the tissues back to the lungs, where it is exhaled. About 70% of carbon dioxide is carried not by hemoglobin, but as bicarbonate ions dissolved in the plasma. This transformation happens with the help of an enzyme called carbonic anhydrase, one of the fastest enzymes in your body. It speeds up the conversion of carbon dioxide and water into carbonic acid, which then splits into bicarbonate and hydrogen ions. This process is so efficient that it happens almost instantly, thousands of times per second. The remaining carbon dioxide, about 20 to 30%, does bind to hemoglobin, but not to the iron atoms. It binds to the protein chains themselves, to amino acids on the surface of the hemoglobin molecule. This form is called carbaminohemoglobin. When hemoglobin releases oxygen in the tissues, it becomes more willing to pick up carbon dioxide. And when it releases carbon dioxide in the lungs, it becomes more willing to pick up oxygen. The two processes are linked, each facilitating the other. It is a molecular dance, a give and take that never stops. A small amount of oxygen, about 1 to 2%, does dissolve directly in the plasma without binding to hemoglobin. This dissolved oxygen is important for certain measurements, and it contributes to the partial pressure of oxygen in the blood, a value that helps drive the diffusion of oxygen into tissues. But most of the work, the real heavy lifting, is done by hemoglobin. Without it, your blood could carry only a fraction of the oxygen your body needs. You would not survive. Hemoglobin is so effective that even at rest, when you're lying still, your blood delivers far more oxygen than your cells actually use. This is a buffer, a safety margin. It means that when you suddenly need more oxygen, when you stand up or climb stairs or simply think hard, your body can meet that demand immediately. There is always reserve, always capacity. This is one of the reasons you can relax. Your body is prepared. It has built-in abundance. The journey of a red blood cell through your body takes about 20 seconds. 20 seconds from the moment it leaves the heart, travels through the lungs to pick up oxygen, returns to the heart, and is pumped out to the body. In those 20 seconds, it will pass through vessels of every size, from the wide aorta, as thick as a garden hose, to capillaries so narrow they are barely visible under a microscope. And then it returns, carrying carbon dioxide, and the cycle begins again. A single red blood cell will make this journey thousands of times in its 4-month life. It is a tireless traveler, never resting, never stopping. The heart, of course, is the engine of this system. It beats about 70 times per minute when you are at rest, a little faster when you are active, a little slower when you sleep. Each beat pushes blood forward, creating the pressure that drives circulation. But the heart does not work alone. The walls of your arteries are elastic, stretching with each pulse and then gently recoiling, helping to smooth the flow of blood. This elasticity is vital. It turns the rhythmic surge of the heartbeat into a more continuous, steady stream. It is a collaboration between muscle and vessel, between force and flexibility. Your blood vessels form a network that, if laid end to end, would stretch for about 60,000 miles. That is more than twice the circumference of the Earth. And yet this entire network fits inside you, branching and rebranching, reaching every corner of your body. The largest vessels are highways, carrying blood quickly over long distances. The smallest, the capillaries, are back roads, winding through tissues, slowing down to allow time for exchange. It is in the capillaries that oxygen leaves the blood and carbon dioxide enters. It is here that the purpose of circulation is fulfilled. The walls of capillaries are thin, just one cell thick, and they are slightly leaky. This is intentional. Fluid, carrying dissolved oxygen and nutrients, seeps out of the capillaries and into the spaces between cells, bathing the tissues in a nourishing liquid. Cells take what they need, and the fluid, now carrying waste products, seeps back into the capillaries or into the lymphatic system, another network that runs parallel to the blood vessels. This slow, gentle exchange is happening everywhere in your body, all the time. It is quiet, invisible, essential. Oxygen is a paradox. It is necessary for life, the fuel that powers nearly every cell in your body. And yet it is also reactive, potentially dangerous. When oxygen is used by cells to produce energy, it can form reactive oxygen species, molecules that can damage proteins, lipids, and DNA. Your body has defenses against this, antioxidants and repair enzymes that neutralize these reactive molecules before they cause harm. But the fact remains, the very thing that gives you life also requires careful management. Your body performs this management automatically, balancing use and protection, risk and reward. It is a delicate equilibrium, maintained without your awareness. The process by which cells use oxygen to produce energy is called cellular respiration. It happens in the mitochondria, tiny structures inside each cell that are often called the powerhouses of the cell. Mitochondria take in oxygen and glucose, and through a series of chemical reactions, they produce ADP, adenosine triphosphate, the molecule that stores and transfers energy. This process is remarkably efficient. From one molecule of glucose, a cell can produce up to 38 molecules of ADP. And the only waste products are carbon dioxide and water. Carbon dioxide is exhaled. Water is reused or excreted. It is a clean, elegant system. Mitochondria themselves are ancient. They were once independent bacteria billions of years ago that entered into a symbiotic relationship with larger cells. Over time, they became permanent residents, losing their independence but gaining a stable home. In return, they provided their host cells with a powerful new way to generate energy. This partnership is one of the most important events in the history of life. It is why complex, multicellular organisms like you exist. It is why you can think, move, dream. Every breath you take honors this ancient alliance. The oxygen you breathe today was not always part of Earth's atmosphere. Billions of years ago, the air was thick with methane, ammonia, and carbon dioxide, but almost no oxygen. The first organisms that learned to harness the energy of sunlight, the ancestors of modern cyanobacteria, began to produce oxygen as a byproduct of photosynthesis. At first, this oxygen was absorbed by iron in the oceans, forming rust that settled to the ocean floor. But eventually, the oceans became saturated, and oxygen began to accumulate in the atmosphere. This event, called the Great Oxidation Event, happened about 2.5 billion years ago. It was a catastrophe for many organisms for whom oxygen was toxic, but it was also an opportunity. It paved the way for new forms of life, forms that could use oxygen to generate energy far more efficiently than ever before. You are the descendant of those survivors. The air you breathe is about 21% oxygen. This concentration is not random. It is the result of billions of years of balance between oxygen-producing photosynthesis and oxygen-consuming respiration and combustion. If the concentration were much higher, wildfires would rage out of control. If it were much lower, complex life would struggle to survive. The current level is a kind of equilibrium maintained by the biosphere as a whole. Every tree, every blade of grass, every microscopic algae in the ocean contributes to this balance. You are breathing the gift of countless living things. Hemoglobin is not unique to humans. It is found in nearly all vertebrates, in fish and frogs and birds and mammals. But it also appears in some invertebrates, in certain worms and mollusks. Though their versions are often different, sometimes carrying oxygen in the fluid outside their cells rather than inside red blood cells. There are also other oxygen-carrying molecules in nature. Some crustaceans and mollusks use hemocyanin, which contains copper instead of iron and gives their blood a blue color. Some marine worms use hemerythrin, which contains iron but in a different configuration, and their blood is violet or pink. Each of these molecules evolved independently in response to the same challenge: how to transport oxygen efficiently. It is a beautiful example of convergent evolution, of life finding similar solutions to the same problem. The study of hemoglobin has taught scientists much about how proteins work, how they fold, how they change shape, how they interact with other molecules. Hemoglobin was one of the first proteins to have its structure determined at the atomic level, a breakthrough that earned Max Perutz and John Kendrew the Nobel Prize in 1962. Their work revealed not just the structure of hemoglobin, but the principles by which all proteins function. It showed that shape and function are inseparable, that the three-dimensional arrangement of atoms determines what a molecule can do. This insight has rippled through all of biology and medicine. There are genetic variations in hemoglobin, mutations that change its structure and function. Some of these are harmful. Sickle cell disease, for example, is caused by a single change in one amino acid in the hemoglobin protein. This small change causes hemoglobin molecules to stick together under low oxygen conditions, distorting red blood cells into a crescent or sickle shape. These misshapen cells are fragile and can block blood vessels, causing pain and damage. And yet, carrying one copy of the sickle cell mutation provides some protection against malaria, a disease caused by a parasite that infects red blood cells, in regions where malaria is common. This mutation has persisted because the benefit of malaria resistance outweighs the cost of carrying one altered gene. It is a reminder that evolution is not about perfection, but about trade-offs, about survival in a particular environment. Another variation is found in people who live at high altitudes, such as in the Andes or the Himalayas. Over many generations, their bodies have adapted to low oxygen levels in ways that go beyond increased BPG. Some have more red blood cells, thicker blood that carries more hemoglobin. Others have hemoglobin that binds oxygen more effectively, or blood vessels that are wider, allowing blood to flow more easily. These adaptations are not conscious choices. They are the result of natural selection, of bodies and genes responding to the environment over thousands of years. They are a testament to the flexibility of life, to the ways in which we are shaped by the places we inhabit. Fetal hemoglobin is different from adult hemoglobin. Before birth, a baby cannot breathe air. It receives oxygen from its mother's blood across the placenta. For this to work, fetal hemoglobin must be able to pull oxygen away from maternal hemoglobin. It does this by binding oxygen more tightly with a higher affinity. This difference is due to a slight change in the protein structure. Fetal hemoglobin has 2 gamma subunits instead of the 2 beta subunits found in adult hemoglobin. This small change shifts the balance, ensuring that oxygen flows from mother to child. After birth, as the baby begins to breathe, the production of fetal hemoglobin gradually decreases and is replaced by adult hemoglobin. The transition is smooth, automatic, guided by genetic switches that respond to the changing needs of the growing body. Oxygen transport is not just about chemistry. It is also about physics, about pressure and diffusion. Oxygen moves from areas of high concentration to areas of low concentration, down what is called a concentration gradient. In the lungs, where oxygen is abundant, it diffuses into the blood. In the tissues, where oxygen is being used up, it diffuses out of the blood. This movement is passive. It requires no energy, no effort. It is driven simply by the difference in concentration, by the natural tendency of molecules to spread out, to equalize. Your body harnesses this tendency, creating the conditions that allow oxygen to flow where it is needed. The partial pressure of oxygen is a way of measuring how much oxygen is present in a gas or a liquid. In the air you breathe, oxygen makes up about 21% of the total pressure. At sea level, this translates to a partial pressure of about 160 millimeters of mercury. In the alveoli of your lungs, after oxygen has mixed with the air already there and some has diffused into the blood, the partial pressure drops to about 100 millimeters of mercury. In the blood leaving the lungs, it is similar, around 100. But in the tissues where oxygen is being consumed, the partial pressure can drop to 40 millimeters of mercury or lower. This gradient, this difference in pressure, is what drives oxygen out of the blood and into the cells. It is a quiet, invisible force, but it is powerful. The amount of oxygen that hemoglobin carries depends on the partial pressure of oxygen in a predictable way, described by the oxygen-hemoglobin dissociation curve. This curve is not a straight line. It is S-shaped or sigmoidal. At low oxygen pressures, hemoglobin holds relatively little oxygen. As pressure increases, hemoglobin begins to bind oxygen more eagerly, and the curve rises steeply. At high pressures, hemoglobin becomes nearly saturated, and the curve flattens out. This shape is important. It means that in the lungs, where oxygen pressure is high, hemoglobin can become fully loaded. And in the tissues, where pressure is lower, hemoglobin releases oxygen readily. The curve is shifted by factors like pH, temperature, and BPG, fine-tuning the system to match the body's needs. When you exercise, your muscles demand more oxygen. Your heart beats faster, pumping more blood per minute. You breathe more deeply and more quickly, bringing more oxygen into your lungs. The capillaries in your muscles open wider, allowing more blood to flow through. Hemoglobin releases oxygen more readily because the muscles are warm, acidic, and full of carbon dioxide. All of these changes happen automatically, coordinated by your nervous system and by chemical signals in the blood. You don't have to think about any of it. Your body knows what to do. Even at rest, your brain consumes about 20% of the oxygen your body uses, despite making up only about 2% of your body weight. Neurons, the cells that transmit signals in your brain, are incredibly active, constantly firing, constantly communicating. This activity requires energy, and energy requires oxygen. The brain is so dependent on a continuous supply of oxygen that even a few minutes without it can cause serious damage. This is why blood flow to the brain is carefully regulated, why the body prioritizes it above almost everything else. It is why you can feel dizzy if you stand up too quickly, as your body works to redirect blood upward against gravity to keep your brain supplied. The color of blood changes depending on how much oxygen it carries. Oxygen-rich blood, the blood in your arteries, is bright red. Oxygen-poor blood, the blood in your veins, is darker, more maroon. This difference is due to the way hemoglobin absorbs light. When oxygen is bound to hemoglobin, the molecule reflects red light more strongly. When oxygen is released, the color shifts. You can see this in your own body. The veins visible beneath your skin often appear blue, but this is an optical illusion caused by the way light penetrates and scatters in your skin. The blood inside those veins is not blue. It is always red, just darker when it carries less oxygen. Blood doping, the practice of increasing the number of red blood cells to enhance athletic performance, works because more red blood cells mean more hemoglobin, and more hemoglobin means more oxygen carrying capacity. Athletes who blood dope can deliver more oxygen to their muscles, delaying fatigue and improving endurance. But this practice is dangerous. Thicker blood flows less easily, increasing the risk of clots, strokes, and heart attacks. It is also considered cheating in most sports, a violation of the principle of fair competition. The body's natural oxygen transport system is already remarkably efficient. Trying to push it beyond its limits carries serious risks. Anemia is a condition in which the blood carries less oxygen than normal, usually because there are too few red blood cells or too little hemoglobin. It can be caused by blood loss, by nutritional deficiencies, especially of iron, by chronic diseases, or by genetic disorders. People with anemia often feel tired, weak, and short of breath because their tissues are not receiving enough oxygen. Treatment depends on the cause, but it often involves increasing iron intake, treating underlying diseases, or in severe cases, blood transfusions. Anemia is a reminder of how finely tuned the oxygen transport system is and how much we depend on it. Carbon monoxide is a dangerous gas because it binds to hemoglobin much more tightly than oxygen does, about 200 times more tightly. When carbon monoxide is present, even in small amounts, it occupies the binding sites on hemoglobin, preventing oxygen from attaching. The result is that the blood cannot carry enough oxygen, and the tissues suffocate even though the person is breathing. Carbon monoxide is colorless, odorless, and tasteless, which makes it especially dangerous. It is produced by incomplete combustion, by car engines, gas heaters, and fires. Detectors and proper ventilation are essential to prevent poisoning. The body can recover if the exposure is not too severe, as carbon monoxide is eventually exhaled and replaced by oxygen, but recovery can take hours or even days. The discovery of oxygen itself is a story of gradual understanding. Several scientists in the 18th century isolated oxygen and studied its properties, including Carl Wilhelm Scheele, Joseph Priestley, and Antoine Lavoisier. Priestley is often credited with the discovery, having produced oxygen by heating mercuric oxide and noting that a candle burned more brightly in the gas. But it was Lavoisier who recognized oxygen as an element and understood its role in combustion and respiration. He named it oxygen, from Greek words meaning acid former, because he mistakenly believed it was a component of all acids. The name stuck, even though the reasoning was flawed. The understanding of how oxygen is transported in the blood came much later, in the 19th and 20th centuries, as scientists unraveled the mysteries of hemoglobin and red blood cells. The spleen, a small organ tucked beneath your ribcage on the left side, plays a role in managing red blood cells. It acts as a filter, removing old, damaged, or misshapen red blood cells from circulation. It also stores a reserve of red blood cells that can be released into the bloodstream when needed, such as during bleeding or intense physical activity. The spleen recycles the iron from old red blood cells, sending it back to the bone marrow to be used in making new hemoglobin. It is a quiet, often overlooked organ, but it is part of the system that keeps your blood healthy and your oxygen transport efficient. Altitude sickness occurs when you ascend to high elevations too quickly, before your body has had time to adapt to the lower oxygen levels. Symptoms include headache, nausea, dizziness, and shortness of breath. The body responds to low oxygen by increasing breathing rate and heart rate, trying to deliver more oxygen to the tissues. Over days or weeks, it also increases the production of red blood cells and BPG, improving oxygen transport. But if you ascend too fast, these adaptations cannot keep pace, and the mismatch between oxygen supply and demand causes discomfort or even danger. The solution is to ascend slowly, allowing time for acclimatization, and to descend if symptoms become severe. It is a reminder that the oxygen transport system, efficient as it is, has limits, and that respecting those limits is essential. The air you breathe out is not devoid of oxygen. Exhaled air still contains about 16% oxygen, compared to the 21% in fresh air. This means you only extract about a quarter of the oxygen available in each breath. This might seem inefficient, but it reflects the balance between the need to remove carbon dioxide and the need to take in oxygen. If you held your breath longer, you could extract more oxygen, but carbon dioxide would build up to dangerous levels. The system is optimized not for maximum extraction, but for continuous, balanced exchange. Oxygen saturation is a measure of how much hemoglobin in your blood is carrying oxygen. It is usually expressed as a percentage. Healthy individuals at sea level typically have oxygen saturations of 95 to 100%. This can be measured non-invasively using a pulse oximeter, a small device that clips onto a fingertip and shines light through the skin. The device detects the color difference between oxygenated and deoxygenated hemoglobin and calculates the saturation. It is a simple, painless test that provides valuable information about how well the respiratory and circulatory systems are working. During the recent global health challenges, pulse oximeters became common household items, helping people monitor their oxygen levels at home. The lymphatic system, though separate from the circulatory system, works in close partnership with it. Lymph vessels collect excess fluid that is leaked out of capillaries, along with waste products and immune cells, and return it to the bloodstream. This prevents tissues from becoming waterlogged and ensures that the balance of fluids in the body is maintained. The lymphatic system does not have a pump like the heart. Instead, it relies on the contraction of muscles and the movement of the body to push lymph through its vessels. This is one reason why physical activity is important for health. It keeps the lymph flowing, supporting the overall function of circulation and oxygen delivery. Blood plasma, the liquid part of blood, makes up about 55% of blood volume. It is mostly water, but it also contains proteins, electrolytes, hormones, and waste products. Plasma serves as the medium in which red blood cells, white blood cells, and platelets are suspended and transported. It also carries dissolved nutrients from the digestive system to the cells, and waste products from the cells to the kidneys and lungs for excretion. Plasma is pale yellow, and when blood is allowed to settle or is spun in a centrifuge, the red blood cells sink to the bottom and the plasma rises to the top. This separation reveals the two main components of blood, the cellular part and the liquid part, each with its own essential functions. The kidneys, though primarily known for filtering waste and producing urine, also play a role in regulating red blood cell production. When the kidneys detect low oxygen levels in the blood, they release a hormone called erythropoietin, or EPO. This hormone travels to the bone marrow and stimulates the production of new red blood cells. It is a feedback loop. Low oxygen triggers EPO release. EPO increases red blood cell production. More red blood cells improve oxygen delivery, and oxygen levels rise. This system allows the body to adjust its oxygen-carrying capacity in response to changing needs or conditions, such as blood loss, high altitude, or chronic lung disease. The liver also contributes to the management of red blood cells. It is involved in the breakdown of old red blood cells, processing the hemoglobin they contain. The iron is salvaged and recycled, while the rest of the hemoglobin is converted into bilirubin a yellow pigment that is excreted in bile and gives feces its brown color. High levels of bilirubin can cause jaundice, a yellowing of the skin and eyes, which can occur in newborns whose livers are not yet fully mature, or in adults with liver disease or excessive red blood cell breakdown. The liver's role in this process is another example of how interconnected the body's systems are, how the transport of oxygen depends not just on the blood, but on the organs that support and regulate it. The journey of oxygen through your blood continues in ways that reveal layer upon layer of quiet intelligence, each mechanism more elegant than the last. Consider for a moment the way your red blood cells navigate the smallest vessels in your body. The capillaries so narrow that these cells must fold and squeeze to pass through. They are remarkably flexible, these disc-shaped travelers, deforming their structure to fit through spaces barely wider than themselves, all while carrying their precious cargo of oxygen molecules bound to hemoglobin. This flexibility is not accidental, but the result of a cell membrane engineered over millions of years to bend without breaking, to yield without tearing, allowing each cell to reach even the most distant tissues. The membrane of a red blood cell is a marvel of biological architecture, composed of a lipid bilayer supported by a flexible protein skeleton beneath. This skeleton, made primarily of a protein called spectrin, forms a network that can stretch and recoil like a microscopic trampoline. When the cell enters a narrow capillary, the spectrin network allows it to elongate, to become almost bullet-shaped, sliding through the tight passage with minimal resistance. Once it emerges into a wider vessel, the cell quietly returns to its biconcave disc shape, ready for the next challenge. This constant shape-shifting happens billions of times throughout the cell's lifespan, a silent dance of adaptation that ensures oxygen reaches every corner of your body. The biconcave shape itself serves multiple purposes beyond flexibility. By being thinner in the center and thicker at the edges, the red blood cell maximizes its surface area relative to its volume, creating more space for oxygen to diffuse in and out. This increased surface area means that oxygen molecules have less distance to travel from the cell membrane to the hemoglobin molecules within, speeding up the entire process of gas exchange. The shape also helps the cell tumble gently as it flows through blood vessels, constantly presenting fresh surfaces to the surrounding plasma, enhancing the efficiency of oxygen pickup and delivery. It is a form perfectly suited to function, sculpted by evolutionary pressures into an optimal design. Within the plasma that surrounds these red blood cells, a small but significant amount of oxygen dissolves directly into the liquid itself. This dissolved oxygen represents only about 2% of the total oxygen carried by blood, but it plays an important role nonetheless. It is this dissolved oxygen that first contacts the tissues, diffusing out of the plasma and into cells before the oxygen bound to hemoglobin is released. The dissolved oxygen also provides the initial signal that triggers hemoglobin to release its own oxygen cargo, creating a smooth, continuous flow of this vital gas from blood to tissue. The plasma acts as a bridge, a medium through which oxygen can move freely, guided by the gentle gradients of concentration that exist throughout your body. Temperature plays a fascinating role in oxygen transport, one that reveals how deeply integrated these systems are with your body's overall state. When your tissues are working hard, generating heat through metabolic activity, that warmth affects hemoglobin's affinity for oxygen. Higher temperatures cause hemoglobin to release oxygen more readily, ensuring that active, heat-producing tissues receive extra oxygen precisely when they need it most. This is another aspect of the Bohr effect, working in concert with pH and carbon dioxide levels to fine-tune oxygen delivery. Your body uses temperature as yet another signal, another way to communicate local needs to the circulating red blood cells passing through. During exercise, this temperature effect becomes particularly important. Your muscles generate significant heat as they contract repeatedly, and this heat spreads through the local tissue, warming the blood flowing through. As the blood temperature rises, hemoglobin loosens its grip on oxygen molecules, allowing them to diffuse more readily into the muscle fibers. This means that the harder a muscle works, the more heat it produces, and the more oxygen it automatically receives, all without any conscious control or complex signaling required. It is a self-regulating system, elegant in its simplicity, responsive in its design. The opposite occurs in cooler parts of your body, such as your skin on a cold day or your extremities in winter. In these areas, the lower temperature causes hemoglobin to hold onto oxygen more tightly, reducing the amount released to the tissues. This might seem counterproductive, but it actually helps conserve oxygen for more vital organs, ensuring that your brain, heart, and other core structures receive adequate supply even when overall circulation to the periphery is reduced. Your body prioritizes survival, quietly shifting resources to where they matter most, guided by the simple physics of temperature and molecular binding. Another compound that influences oxygen transport is a molecule called 2,3-bisphosphoglycerate, often abbreviated as 2,3-BPG. This small organic molecule is produced within red blood cells as a byproduct of glucose metabolism, and it has a remarkable effect on hemoglobin. When 2,3-BPG binds to hemoglobin, it stabilizes the deoxygenated form of the protein, making it slightly less eager to hold onto oxygen. This might sound like a disadvantage, but it actually helps ensure that oxygen is released to the tissues rather than remaining bound to hemoglobin as it circulates. The presence of 2,3-BPG shifts the oxygen-hemoglobin dissociation curve to the right, favoring oxygen unloading in the capillaries. Your body adjusts the levels of 2,3-BPG in response to various conditions, particularly those involving chronic low oxygen availability. If you travel to high altitude and remain there for days or weeks, your red blood cells gradually increase their production of 2,3-BPG, helping to compensate for the lower oxygen pressure in the air. With more 2,3-BPG present, hemoglobin releases oxygen more readily to your tissues, partially offsetting the reduced oxygen loading that occurs in your lungs. This adaptation takes time, developing over days rather than minutes, but it represents one of the ways your body can adjust to sustained changes in oxygen availability. It is a quiet recalibration happening deep within your cells, preparing you for a different environment. Interestingly, stored blood used for transfusions gradually loses its 2,3-BPG over time, as the red blood cells sit in refrigerated storage without the metabolic machinery of a living body to replenish it. This means that transfused blood may initially be less effective at releasing oxygen to tissues, though the recipient's body typically restores normal 2,3-BPG levels within hours to days after transfusion. It is a reminder that blood is not a static fluid, but a living tissue, constantly active and responsive, even when it appears to be simply flowing quietly through vessels. The spleen plays a subtle but important role in maintaining the quality of your circulating red blood cells. This organ, nestled beneath your rib cage on the left side, acts as a filter and quality control center for blood. As red blood cells age over their 4-month lifespan, they become less flexible, their membranes stiffen, and they struggle to navigate the narrow passages of capillaries. The spleen detects these aging cells and removes them from circulation, breaking them down and recycling their components. This process ensures that your blood contains primarily healthy, flexible red blood cells capable of efficiently transporting oxygen. The spleen works quietly, without fanfare, maintaining the quality of your blood day after day. The iron from old red blood cells is carefully salvaged and recycled, a process that speaks to the body's remarkable efficiency. Iron is relatively scarce in the diet and precious to the body. So rather than excreting it, your body recovers the iron from degraded hemoglobin and transports it back to the bone marrow, where it can be incorporated into new hemoglobin molecules for new red blood cells. This recycling system is so efficient that most of the iron in your body circulates continuously through this loop, being used, reclaimed, and reused throughout your lifetime. It is a closed cycle, a sustainable system that minimizes waste and maximizes resource utilization. The protein that transports iron through your bloodstream is called transferrin, a molecule specifically designed to bind iron atoms and carry them safely through the plasma. Free iron is potentially toxic, capable of generating harmful reactive oxygen species, so your body keeps it carefully sequestered, bound to proteins at all times. Transferrin shuttles iron from the spleen and other sites of red blood cell breakdown to the bone marrow and other tissues that need it, ensuring a steady supply for hemoglobin synthesis. This transport system operates continuously, moving iron atoms from old cells to new ones in an endless cycle of renewal. In the bone marrow, developing red blood cells, called erythroblasts, are busy assembling new hemoglobin molecules. These immature cells contain nuclei and the full complement of cellular machinery needed for protein synthesis. They produce hemoglobin at a remarkable rate, filling their interiors with this oxygen-carrying protein. As they mature, something extraordinary happens. They expel their nuclei, along with most of their other organelles, creating more internal space for hemoglobin. This self-reduction transforms them into reticulocytes, cells that still contain some residual RNA and ribosomes, but are nearly mature. After a day or two in circulation, even these remnants disappear, and the cell becomes a fully mature red blood cell, a simple sac filled almost entirely with hemoglobin and the enzymes needed to maintain it. The loss of a nucleus means that mature red blood cells cannot produce new proteins or repair significant damage to their existing ones. This limitation defines their lifespan, as accumulated wear and tear gradually degrades their hemoglobin and membrane proteins over the course of about 120 days. Without the ability to synthesize new components, the cells eventually reach a point where they can no longer function effectively, and they are removed by the spleen. It is a trade-off, sacrificing longevity and adaptability for maximum oxygen-carrying capacity, a design choice that prioritizes the cell's primary function above all else. The rate at which your bone marrow produces new red blood cells is carefully regulated by a hormone called erythropoietin, or EPO. This hormone is produced primarily by the kidneys, which monitor the oxygen levels in blood passing through them. When the kidneys detect that oxygen levels are lower than optimal, they increase their production of EPO, sending this chemical signal through the bloodstream to the bone marrow. The marrow responds by accelerating red blood cell production, increasing the number of oxygen carriers in circulation. This feedback loop ensures that your blood always contains an appropriate number of red blood cells for your current needs, adjusting production up or down as conditions change. At high altitude, where oxygen is scarce, your kidneys release more EPO, stimulating the production of additional red blood cells over the course of days to weeks. This increase in red blood cell count, called polycythemia, helps compensate for the reduced oxygen availability by providing more hemoglobin molecules to capture whatever oxygen is present. Athletes sometimes train at high altitude specifically to trigger this response, hoping that the increased red blood cell count will enhance their performance when they return to sea level. The body's natural response to altitude becomes a tool for optimization, though the effect is temporary, as red blood cell counts gradually return to normal once the altitude stimulus is removed. Certain medical conditions can disrupt this finely tuned system, leading to either too few or too many red blood cells. Anemia, a condition characterized by insufficient red blood cells or hemoglobin, reduces the oxygen-carrying capacity of blood, leaving tissues undersupplied and causing fatigue, weakness, and shortness of breath. Anemia can result from many causes, including iron deficiency, vitamin deficiencies, chronic diseases, or genetic disorders affecting hemoglobin production. Each type requires different treatment, but all share the common feature of impaired oxygen transport, a reminder of how dependent we are on this elegant system functioning properly. Conversely, polycythemia vera, a condition in which the bone marrow produces too many red blood cells, can make blood thick and sluggish, increasing the risk of clots and strokes. Even too much of a good thing can cause problems when it disrupts the careful balance your body maintains. The ideal number of red blood cells is not the maximum possible, but rather the amount that optimizes oxygen delivery while maintaining appropriate blood viscosity and flow characteristics. Your body seeks balance, not extremes, and this is in so many other physiological systems. The color of blood itself offers clues about oxygen transport, though the reality is more nuanced than often portrayed. Oxygen-rich blood, such as that flowing through arteries after leaving the lungs, is bright red due to the way oxygenated hemoglobin absorbs and reflects light. Oxygen-poor blood, returning through veins from the tissues, is darker red, sometimes appearing almost maroon. The common misconception that deoxygenated blood is blue likely arises from the appearance of veins through skin, where light scattering and absorption by tissue layers create a bluish appearance. But blood itself is always some shade of red, the exact hue determined by how much oxygen is bound to its hemoglobin. This color change is so reliable that medical devices called pulse oximeters use it to measure blood oxygen levels non-invasively. By shining light through a fingertip or earlobe and measuring how much light is absorbed at different wavelengths, these devices can determine what percentage of hemoglobin is carrying oxygen. It is a clever application of the optical properties of hemoglobin, turning a molecular characteristic into a practical diagnostic tool. The device reads the color of your blood without needing to draw a single drop, translating light absorption into a number that tells clinicians whether your oxygen transport system is functioning adequately. Carbon monoxide poses a serious threat to oxygen transport because of its molecular similarity to oxygen and its much stronger affinity for hemoglobin. When carbon monoxide is present, even in small amounts, it binds to hemoglobin far more readily than oxygen does, occupying binding sites and preventing oxygen from attaching. Worse, carbon monoxide binding changes the shape of hemoglobin in a way that makes it even harder for oxygen to bind to the remaining sites, amplifying the problem. A person exposed to carbon monoxide may have blood that is fully saturated with gas molecules, but those molecules are carbon monoxide rather than oxygen, rendering the blood unable to deliver the oxygen that tissues desperately need. The treatment for carbon monoxide poisoning involves breathing pure oxygen or, in severe cases, hyperbaric oxygen at high pressure. The goal is to flood the system with oxygen molecules, using sheer numbers to compete with carbon monoxide for hemoglobin binding sites and gradually displace the toxic gas. This process takes time, as carbon monoxide releases from hemoglobin slowly, but with sustained oxygen therapy, the hemoglobin can eventually be cleared and restored to its proper function. It is a dramatic example of how vulnerable the oxygen transport system is to disruption and how dependent we are on hemoglobin binding the right molecule. Nitric oxide, despite its name suggesting a toxic gas, actually plays beneficial roles in oxygen delivery and blood flow regulation. This simple molecule, produced by cells lining blood vessels, causes the smooth muscle and vessel walls to relax, widening the vessels and increasing blood flow. When tissues need more oxygen, they can trigger local nitric oxide release, which dilates nearby blood vessels, bringing more blood and thus more oxygen to the area. Hemoglobin itself interacts with nitric oxide in complex ways, sometimes binding it and transporting it to distant locations, sometimes releasing compounds that generate it locally. These interactions help coordinate blood flow with oxygen demand, ensuring that circulation adjusts dynamically to meet changing needs. The relationship between hemoglobin and nitric oxide is still being fully understood, but it appears that hemoglobin may carry nitric oxide or related compounds attached to specific amino acids, releasing them when oxygen is unloaded. This would create a coordinated response where oxygen delivery and blood vessel dilation happen together, each enhancing the other. It is another layer of sophistication in the oxygen transport system, another way that multiple mechanisms work in concert to optimize performance. The more we learn about these systems, the more elegant and interconnected they appear. Myoglobin, a protein found in muscle tissue, acts as a local oxygen storage system, complementing the oxygen delivery provided by hemoglobin in blood. Myoglobin is similar in structure to a single subunit of hemoglobin, but exists as a standalone protein rather than a 4-subunit complex. It has a very high affinity for oxygen, meaning it holds onto oxygen tightly and only releases it when oxygen levels drop very low. This makes myoglobin an excellent oxygen reserve, capturing oxygen from hemoglobin when blood oxygen levels are normal and releasing it only when the muscle is working so hard that blood supply cannot keep up with demand. In muscles that must sustain activity for extended periods, such as the heart or the postural muscles that keep you upright, myoglobin is particularly abundant. The high concentration of myoglobin in these muscles gives them a darker red color, which is why different cuts of meat vary in color depending on a muscle's function. Muscles used for sustained activity are darker because they contain more myoglobin, more oxygen storage capacity, more ability to continue working even when blood flow is temporarily reduced. It is a visible marker of metabolic strategy, a protein's abundance revealed in the color of tissue. Marine mammals like whales and seals have extraordinarily high concentrations of myoglobin in their muscles, far exceeding anything found in terrestrial mammals. This adaptation allows them to store large amounts of oxygen in their muscles, supplementing the oxygen carried in their blood and enabling them to hold their breath for extended periods during deep dives. A whale's muscles may be almost black with myoglobin, packed with oxygen-storing protein that sustains the animal through long minutes or even hours underwater. It is the same basic molecule found in human muscle, but expressed in such abundance that it transforms the animal's capabilities, extending breath hold times far beyond what we can achieve. The evolution of oxygen transport systems reveals a long history of life adapting to changing atmospheric conditions. Early in Earth's history, oxygen was scarce, and organisms relied on simple diffusion to move this gas through their bodies. As oxygen levels rose and life forms grew larger and more complex, diffusion alone became insufficient, and specialized transport systems evolved. Hemoglobin and similar proteins appear across diverse branches of the tree of life, from vertebrates to invertebrates, even in some plants and bacteria, suggesting that the basic strategy of using metal-containing proteins to bind and transport oxygen has been discovered and refined many times independently. The hemoglobin in your blood shares a common evolutionary ancestor with the oxygen-carrying proteins found in insects, mollusks, and many other organisms, even though the specific proteins differ in structure and in the metal they use. Some invertebrates use hemocyanin, a copper-based protein that makes their blood blue when oxygenated, rather than red. Others use hemerythrin, an iron-based protein that nonetheless differs significantly from hemoglobin. Each of these proteins represents a different solution to the same problem: how to efficiently transport oxygen through a body too large for diffusion alone. The convergence on metal-containing proteins suggests that this is one of the best available solutions, a design that chemistry and physics favor. The specific choice of iron and hemoglobin may relate to iron's abundance in Earth's crust and oceans, making it readily available as life evolved. Iron's chemistry, with its ability to reversibly bind oxygen without being permanently oxidized under the right conditions, makes it particularly suitable for this role. Copper, used in hemocyanin, offers similar properties but is less abundant. The proteins that evolve to use each metal differ in their oxygen-binding characteristics, with hemoglobin generally providing more efficient oxygen loading and unloading under the conditions found in vertebrate bodies. It is a solution shaped by both chemical possibility and evolutionary history, a path chosen long ago and refined over hundreds of millions of years. Fetal hemoglobin demonstrates how evolution fine-tunes proteins for specific circumstances. Before birth, a developing fetus cannot breathe air and instead receives oxygen from maternal blood across the placenta. For this transfer to work efficiently, fetal hemoglobin must be able to extract oxygen from maternal hemoglobin, pulling it across the placental barrier. Fetal hemoglobin achieves this by having a higher oxygen affinity than adult hemoglobin, allowing it to steal oxygen from the mother's blood. This difference in affinity creates a gradient that drives oxygen transfer from mother to fetus, ensuring the developing baby receives adequate oxygen even though it never takes a breath. The molecular basis for this difference lies in the gamma chains that replace the beta chains in fetal hemoglobin. These gamma chains interact differently with 2,3-BPG, binding it less strongly than beta chains do. Since 2,3-BPG normally reduces hemoglobin's oxygen affinity, the weaker binding in fetal hemoglobin means that this effect is diminished, leaving fetal hemoglobin with a higher affinity for oxygen. It is a subtle change, just a different version of one protein subunit, but it has profound functional consequences, enabling the oxygen transfer that sustains fetal development. After birth, production of gamma chains decreases and beta chains take over, gradually converting the infant's hemoglobin to the adult form as the baby begins breathing air. Some genetic variations in hemoglobin provide advantages under certain conditions, while causing problems under others. Sickle cell trait, in which a person carries one copy of the sickle cell mutation and one normal copy, provides significant resistance to malaria. The malaria parasite has difficulty completing its life cycle in red blood cells that contain sickle hemoglobin, giving carriers of the trait a survival advantage in regions where malaria is common. This explains why the sickle cell mutation is most common in populations from areas with endemic malaria, despite the serious health problems it causes when present in two copies. It is a stark example of evolutionary trade-offs, where a mutation that is harmful in one context provides benefits in another. The fact that such variations persist in human populations reminds us that evolution does not produce perfect solutions, but rather workable compromises shaped by the specific challenges organisms face. The oxygen transport system, elegant as it is, carries the marks of its evolutionary history, including variations that reflect adaptations to local conditions, past environmental pressures, and the constraints of working with existing biological machinery. It is a system that works remarkably well most of the time, but it is not immune to disruption, not infinitely adaptable, not designed from scratch for optimal performance. It is evolved, cobbled together from available materials, refined over time, good enough to have carried our ancestors through millions of years and billions of breaths. The journey through oxygen transport continues softly, and we find ourselves now exploring the delicate balance between oxygen delivery and the body's ever-changing needs. Every breath you take, every heartbeat that pulses through your chest, adjusts itself to match the demands of the moment. When you climb a flight of stairs, your muscles call out for more oxygen, and your body responds with quiet precision, increasing both breathing rate and heart rate in a coordinated dance. This responsiveness happens without conscious thought, a gift of ancient regulatory systems that have been refined over millions of years. The carotid bodies, small clusters of specialized cells nestled near the branching of your carotid arteries in your neck, serve as oxygen sensors for your entire body. They are remarkably sensitive, detecting even slight drops in blood oxygen levels. When oxygen decreases, these cells send urgent signals to the brainstem, prompting deeper, faster breathing. This reflex operates continuously, adjusting your respiratory rhythm to maintain oxygen homeostasis. Even as you rest here, drifting towards sleep, these sentinels remain vigilant, ensuring that every cell receives the oxygen it needs. Similarly, the aortic bodies, located near the arch of your aorta, monitor oxygen levels in the blood leaving your heart. Together with the carotid bodies, they form a surveillance network that keeps your breathing synchronized with your body's metabolic state. These chemoreceptors respond not only to low oxygen, but also to rising carbon dioxide and increasing acidity in the blood. The system is elegantly redundant, with multiple sensors providing overlapping information, ensuring that no dangerous change goes unnoticed. Within the brainstem itself, in a region called the medulla oblongata, lies the respiratory control center. This ancient part of the brain integrates signals from the carotid and aortic bodies, from stretch receptors in the lungs, and from chemical sensors within the brain itself. It orchestrates the rhythm of breathing, sending rhythmic commands to the diaphragm and intercostal muscles. The pattern it creates is not rigid but fluid, adapting moment by moment to your body's needs. When you sleep, this center adjusts your breathing to a slower, deeper pattern, conserving energy while maintaining adequate oxygen supply. The blood-brain barrier, a selective filter formed by tightly joined cells lining the brain's blood vessels, carefully controls what enters the brain's delicate environment. Oxygen, being small and lipid-soluble, passes through easily, ensuring that the brain's enormous energy demands are met. The brain, though only about 2% of your body weight, consumes roughly 20% of your oxygen supply. This disproportionate need reflects the brain's constant activity, the ceaseless firing of neurons, the maintenance of electrical gradients, and the synthesis of neurotransmitters. Even in sleep, the brain remains metabolically active, processing memories and regulating bodily functions. Red blood cells have a lifespan of about 120 days, a finite existence shaped by the wear and tear of their journey. As they age, their membranes become less flexible, their enzymes less efficient, and their ability to navigate narrow capillaries diminishes. The spleen, ever watchful, identifies these aging cells and removes them from circulation. This process is gentle and continuous, with about 1% of your red blood cells replaced each day. The turnover is so gradual that you never notice it, yet it ensures that your blood remains fresh and efficient. When old red blood cells are broken down, their components are carefully salvaged. The iron from hemoglobin is extracted and transported back to the bone marrow, where it will be incorporated into new hemoglobin molecules. This recycling is remarkably efficient, with very little iron lost from the body under normal circumstances. The heme portion of hemoglobin, minus its iron, is converted into bilirubin, a yellow pigment that travels to the liver. There, it is processed and eventually excreted in bile, giving feces their characteristic brown color. This quiet recycling system has operated within you since birth, a circular economy at the cellular level. The bone marrow, nestled within the hollow spaces of your bones, is the birthplace of red blood cells. Here, in a soft, spongy tissue, stem cells divide and differentiate, following a carefully choreographed developmental pathway. A single stem cell can give rise to millions of red blood cells over time. The process, called erythropoiesis, takes about 7 days from the first committed precursor to a mature red blood cell ready to enter circulation. During this time, the developing cell synthesizes hemoglobin ejects its nucleus and other organelles, and reshapes itself into the characteristic biconcave disc. Erythropoietin, the hormone that stimulates red blood cell production, is produced primarily by the kidneys. These organs, filtering your blood continuously, are exquisitely sensitive to oxygen levels. When the kidneys detect low oxygen, specialized cells release erythropoietin into the bloodstream. This hormone travels to the bone marrow, where it binds to receptors on developing red blood cells, promoting their survival and maturation. The system operates as a negative feedback loop. As red blood cell numbers increase and oxygen delivery improves, erythropoietin production decreases. This elegant mechanism maintains red blood cell counts within a narrow, healthy range. At high altitudes, where the air is thin and oxygen partial pressure is low, the body must adapt to chronic oxygen scarcity. Within hours of ascending to high elevation, erythropoietin levels begin to rise. Over days and weeks, red blood cell production increases, boosting the blood's oxygen-carrying capacity. People who live at high altitudes for generations develop additional adaptations, including larger lung capacities and more efficient oxygen extraction. The Tibetan people, for instance, have genetic variations that allow them to thrive at elevations where lowlanders struggle. These adaptations remind us that oxygen transport is not fixed but flexible, shaped by both immediate circumstances and evolutionary history. During pregnancy, a woman's blood volume increases by about 40 to 50%, a remarkable expansion that supports both her own needs and those of the developing fetus. Red blood cell production increases as well, though not quite as much as plasma volume, resulting in a mild dilution of the blood. This physiological anemia of pregnancy is normal and beneficial, reducing blood viscosity and improving flow through the placenta. The fetus, meanwhile, develops its own red blood cells, initially in the yolk sac, then in the liver, and finally in the bone marrow. Fetal hemoglobin, with its higher oxygen affinity, ensures that oxygen flows from maternal blood to fetal blood across the placental barrier. The placenta itself is a marvel of oxygen exchange, a temporary organ that exists only during pregnancy. Its structure maximizes contact between maternal and fetal blood without allowing the two to mix. Maternal blood flows into spaces called intervillous lacunae, bathing finger-like projections called chorionic villi that contain fetal blood vessels. Oxygen diffuses from maternal blood across the thin barrier of the villus and into fetal blood. Carbon dioxide travels in the opposite direction. This exchange happens continuously, sustaining the fetus throughout its development. The placenta is both barrier and bridge, protecting the fetus while connecting it to the mother's circulatory system. After birth, the newborn takes its first breath, and the lungs, previously collapsed and fluid-filled, expand and fill with air. This first breath requires significant effort, overcoming the surface tension of the fluid-lined alveoli. Surfactant, produced by specialized cells in the lungs during late pregnancy, reduces this surface tension, making breathing easier. With the first breaths, oxygen enters the blood, and the newborn circulatory system undergoes rapid changes. Fetal shunts, pathways that allowed blood to bypass the non-functional fetal lungs, close within hours to days. The baby begins producing adult hemoglobin, gradually replacing fetal hemoglobin over the first months of life. Oxygen toxicity, though less commonly discussed than oxygen deprivation, is a real phenomenon. Breathing pure oxygen at high pressures, such as during deep-sea diving or hyperbaric oxygen therapy, can lead to the production of excessive reactive oxygen species. These molecules, including superoxide and hydrogen peroxide, can damage cellular components, particularly lipids and cell membranes and proteins involved in metabolism. The lungs and central nervous system are especially vulnerable. Symptoms of oxygen toxicity can include visual disturbances, nausea, twitching, and, in severe cases, seizures. The body's antioxidant defenses, including enzymes like superoxide dismutase and catalase, normally protect against reactive oxygen species, but they can be overwhelmed by extreme oxygen exposure. Free radicals, despite their potential for harm, also play important roles in normal physiology. White blood cells, for instance, use reactive oxygen species to kill bacteria and other pathogens. The controlled production of these molecules is part of the immune response. Similarly, reactive oxygen species serve as signaling molecules, influencing processes like blood vessel dilation and gene expression. The key is balance. Too few reactive oxygen species, and the immune system falters. Too many, and cellular damage accumulates. Antioxidants from the diet, including vitamins C and E, support the body's natural defenses, helping to maintain this delicate equilibrium. Methemoglobin is an altered form of hemoglobin in which the iron atom has been oxidized from the ferrous state to the ferric state. In this form, hemoglobin cannot bind oxygen. Normally, a small amount of methemoglobin is always present in the blood, but an enzyme called methemoglobin reductase continuously converts it back to functional hemoglobin. Certain drugs, chemicals, and genetic conditions can increase methemoglobin levels, leading to a condition called methemoglobinemia. When methemoglobin levels rise significantly, the blood's oxygen-carrying capacity drops, and the skin may take on a bluish tint. Treatment involves administering methylene blue, a compound that helps reduce methemoglobin back to its functional form. The color of blood, that deep crimson hue, comes from hemoglobin. Oxygenated blood, rich in oxyhemoglobin, is bright red, the color you see when you cut your finger and capillary blood wells up. Deoxygenated blood, found in veins returning to the heart, is darker, a deeper maroon. The common misconception that venous blood is blue likely arises from the appearance of veins beneath the skin, where light scattering and absorption make them appear bluish. In reality, all human blood is some shade of red, though the exact hue depends on how much oxygen it carries. Some animals have evolved entirely different oxygen transport systems. Horseshoe crabs, ancient creatures that have survived relatively unchanged for hundreds of millions of years, use hemocyanin instead of hemoglobin. Hemocyanin contains copper rather than iron, and when oxygenated, it gives the blood a blue color. This protein is less efficient than hemoglobin at binding oxygen, but it works well in the cold, oxygen-rich waters where horseshoe crabs live. Similarly, some Antarctic fish have evolved hemoglobin with altered properties suited to icy waters, and a few species have lost hemoglobin entirely, relying instead on oxygen dissolved directly in their plasma. These variations illustrate the diverse evolutionary solutions to the challenge of oxygen transport. Diving mammals, such as seals, whales, and dolphins, have extraordinary adaptations for managing oxygen during long dives. They have higher concentrations of hemoglobin in their blood and more myoglobin in their muscles than land mammals, allowing them to store more oxygen. During a dive, their heart rate slows dramatically, and blood flow is redirected away from less essential tissues toward the brain and heart. This dive reflex conserves oxygen, extending the time they can remain submerged. Some species can dive for over an hour, descending to depths where pressure is immense and light does not penetrate. When they surface, they breathe rapidly, replenishing oxygen stores before diving again. The human dive reflex, a vestige of our aquatic ancestry, still exists, though in a milder form. When your face is immersed in cold water, your heart rate slows, and blood vessels in your extremities constrict, redirecting blood to vital organs. This reflex is most pronounced in infants and young children, a protective mechanism that may reduce oxygen consumption during accidental submersion. Competitive freedivers train to enhance this reflex, enabling them to hold their breath for several minutes and dive to remarkable depths without breathing apparatus. Their bodies adapt over time, with increased lung capacity, more efficient oxygen use, and greater tolerance for carbon dioxide buildup. Carbon dioxide, though often considered merely a waste product, is essential for regulating breathing. The respiratory control center in the brainstem is more sensitive to carbon dioxide levels than to oxygen levels. When carbon dioxide rises, the blood becomes more acidic, and this acidity is detected by chemoreceptors. The response is an increase in breathing rate and depth, expelling excess carbon dioxide and restoring pH balance. This system means that under normal conditions, you breathe not because you're running out of oxygen, but because carbon dioxide is accumulating. It is a subtle but crucial distinction, one that shapes how your body manages gas exchange. Hyperventilation, rapid and deep breathing, can lower carbon dioxide levels too much, leading to a condition called respiratory alkalosis. As carbon dioxide drops, blood pH rises, becoming more alkaline. This can cause dizziness, tingling in the fingers and lips, and even muscle spasms. Paradoxically, hyperventilation does not significantly increase blood oxygen levels, because hemoglobin is already nearly saturated under normal conditions. The main effect is on carbon dioxide, and the resulting alkalosis can reduce oxygen delivery to tissues by shifting the oxygen-hemoglobin dissociation curve to the left. This is why controlled, steady breathing is more effective than rapid, shallow breaths, even during exertion. Breath holding, on the other hand, allows carbon dioxide to accumulate. The urge to breathe that you feel during breath holding is driven primarily by rising carbon dioxide, not by falling oxygen. Trained individuals can override this urge to some extent, but eventually, the drive becomes overwhelming. If breath holding continues too long, oxygen levels can drop to dangerous levels, potentially causing loss of consciousness. This is why hyperventilating before breath holding, a practice called shallow water blackout when done before underwater swimming, is dangerous. It lowers carbon dioxide without significantly increasing oxygen stores, delaying the urge to breathe until oxygen is critically low. The oxygen we breathe today is a product of photosynthesis, a process carried out by plants, algae, and cyanobacteria. These organisms capture light energy and use it to split water molecules, releasing oxygen as a byproduct. Over billions of years, photosynthetic organisms have transformed Earth's atmosphere from one that was essentially oxygen-free to one that is about 21% oxygen. This oxygen accumulation, known as the Great Oxidation Event, occurred roughly 2.5 billion years ago, driven largely by cyanobacteria. It was a turning point in Earth's history, enabling the evolution of complex, oxygen-breathing life. Before the Great Oxidation Event, most life was anaerobic, thriving in environments without oxygen. Oxygen, in fact, was toxic to many of these early organisms. As oxygen levels rose, anaerobic life retreated to oxygen-free niches, such as deep sediments and the guts of animals. Meanwhile, some organisms evolved mechanisms to tolerate and eventually exploit oxygen, using it in cellular respiration to extract far more energy from nutrients than anaerobic processes could provide. Aerobic respiration, the process that occurs in mitochondria, yields up to 38 molecules of ADP from a single glucose molecule, compared to just 2 from anaerobic glycolysis. This energy windfall powered the evolution of larger, more complex organisms. Mitochondria, the organelles where aerobic respiration occurs, have their own fascinating origin story. They are descended from ancient bacteria that were engulfed by a larger cell in a symbiotic relationship that began over 1 billion years ago. These bacteria brought with them the ability to use oxygen to generate energy efficiently. Over time, they became integrated into the host cell, losing many of their genes, but retaining those essential for energy production. Today, mitochondria still carry a small, circular genome, a remnant of their bacterial ancestry. They reproduce independently within cells, dividing much like bacteria. Every mitochondrion in your body is a living link to this ancient partnership. The electron transport chain, located in the inner membrane of mitochondria, is the final stage of aerobic respiration. Here, electrons from nutrients are passed along a series of protein complexes, releasing energy that is used to pump protons across the membrane. This creates a gradient, a difference in proton concentration, which drives the synthesis of ADP by an enzyme called ADP synthase. Oxygen serves as the final electron acceptor, combining with electrons and protons to form water. This process is continuous and efficient, generating the vast majority of the ADP that powers your cells. Without oxygen, the electron transport chain halts, and ADP production plummets. ADP, adenosine triphosphate, is the universal energy currency of cells. It is a small molecule consisting of an adenine base, a ribose sugar, and 3 phosphate groups. The bonds between the phosphate groups store energy, which is released when ADP is broken down into ADP, adenosine diphosphate, and a free phosphate. This energy powers countless cellular processes: muscle contraction, nerve impulse transmission, protein synthesis, active transport across membranes, and much more. Your body produces and consumes an astonishing amount of ADP each day, roughly your own body weight, though the same molecules are recycled over and over. Muscle cells, especially those in the heart, are packed with mitochondria, reflecting their high energy demands. Cardiac muscle cells can contain thousands of mitochondria, occupying up to 40% of the cell's volume. This abundance ensures a steady supply of ADP to sustain the heart's relentless contractions. Skeletal muscles, too, vary in mitochondrial content depending on their function. Slow-twitch fibers, used for endurance activities, are rich in mitochondria and rely heavily on aerobic respiration. Fast-twitch fibers, used for short bursts of power, have fewer mitochondria and depend more on anaerobic glycolysis. This is why sprinters and marathon runners have different muscle compositions, each optimized for their specific demands. Lactic acid, often blamed for muscle soreness, is actually a product of anaerobic glycolysis. When muscles work intensely and oxygen delivery cannot keep pace with demand, they switch to anaerobic metabolism, breaking down glucose without oxygen. This process is fast but inefficient, producing lactic acid as a byproduct. Lactic acid can be converted back to glucose in the liver, a process called the Cori cycle, or it can be used as fuel by the heart and other tissues. The burning sensation during intense exercise is due to lactic acid and associated acidity, but the delayed soreness you feel a day or two later, known as delayed onset muscle soreness, is caused by microscopic damage to muscle fibers and the inflammation that follows. The liver, a large organ in your upper abdomen, plays a central role in metabolism and oxygen management. It processes nutrients absorbed from the digestive tract, stores glucose as glycogen, and releases it when blood sugar drops. The liver also detoxifies harmful substances, synthesizes proteins, and produces bile. Its blood supply is unique. It receives oxygen-rich blood from the hepatic artery and nutrient-rich, but oxygen-poor blood from the portal vein, which drains the intestines. This dual supply allows the liver to perform its diverse functions. Hepatocytes, the main liver cells, are metabolically active and rich in mitochondria, reflecting the liver's role as the body's metabolic hub. The kidneys, too, are highly vascular organs, receiving about 20% of the heart's output despite being only about 1% of body weight. They filter blood continuously, removing waste products and excess substances while retaining essential nutrients and water. The filtration process is energy-intensive, requiring a steady supply of oxygen and ATP. Each kidney contains about 1 million functional units called nephrons, which filter blood, reabsorb needed substances, and secrete waste into urine. The kidneys also regulate electrolyte balance, blood pressure, and red blood cell production through erythropoietin secretion. Their work is quiet and continuous, essential for maintaining the internal environment that allows oxygen transport and cellular respiration to proceed smoothly. The skin, your body's largest organ, also participates in gas exchange, though to a much smaller extent than the lungs. A tiny fraction of oxygen and carbon dioxide diffuses directly through the skin, especially in areas where the skin is thin and blood vessels are close to the surface. In some amphibians, such as frogs, cutaneous respiration is significant, supplementing or even replacing lung breathing. In humans, the contribution is negligible, but it is a reminder that the boundary between body and environment is not absolute. Every surface in contact with air is a potential site of gas exchange, though the lungs remain by far the most efficient. Oxygen's role extends beyond energy production. It is involved in the synthesis of collagen, the structural protein that forms the framework of skin, bones, tendons, and blood vessels. Enzymes that build collagen require oxygen to function, which is why adequate oxygen delivery is essential for wound healing. When tissues are injured, blood flow increases, bringing oxygen and nutrients to support repair. New blood vessels sprout, a process called angiogenesis, guided by signals released by oxygen-starved cells. Fibroblasts, the cells that produce collagen, proliferate and lay down new extracellular matrix. Over time, the wound closes and strengthens, a process that depends on the continuous, quiet work of oxygen transport. Angiogenesis, the formation of new blood vessels, is tightly regulated. It occurs during growth, wound healing, and in response to exercise. When tissues experience low oxygen, they release a protein called hypoxia-inducible factor, or HIF. HIF activates genes that promote blood vessel growth, increasing oxygen delivery to the affected area. This same mechanism, however, can be hijacked by tumors, which stimulate angiogenesis to support their rapid growth. Cancer researchers study angiogenesis intensely, seeking ways to block tumor blood vessel formation and starve tumors of oxygen. Understanding oxygen transport and its regulation thus has implications far beyond normal physiology, touching on disease and therapy. Exercise training enhances the body's oxygen transport and utilization systems in multiple ways. The heart grows stronger, pumping more blood with each beat. Capillary density in muscles increases, shortening the distance oxygen must diffuse to reach cells. Mitochondria proliferate, boosting the capacity for aerobic respiration. These adaptations improve endurance, allowing trained individuals to sustain higher levels of activity with less fatigue. The changes occur gradually, over weeks and months, in response to the repeated stimulus of exercise. They are reversible, too. Without continued training, the adaptations fade. This plasticity reflects the body's ability to match its structure and function to the demands placed upon it. Altitude training, used by many endurance athletes, exploits the body's adaptive response to low oxygen. By training at high elevations, athletes stimulate increased red blood cell production and other physiological changes that enhance oxygen delivery. When they return to sea level, these adaptations persist for a time, providing a performance advantage. Some athletes use live high, train low protocols, living at altitude to gain the hematological benefits, while training at lower elevations where oxygen availability supports high-intensity workouts. The strategy is complex and must be carefully managed, but it illustrates the body's remarkable flexibility in responding to environmental challenges. Sleep, that nightly retreat into unconsciousness, is a time when oxygen transport continues unabated, though the body's demands shift. Metabolic rate decreases, and breathing slows and deepens. The brain remains active, cycling through stages of sleep, each with distinct patterns of electrical activity. During REM sleep, when dreaming is most vivid, brain oxygen consumption may actually increase, approaching waking levels. The respiratory control center adjusts breathing to maintain stable blood gases, even as consciousness fades. For most people, this regulation is seamless, but in some, sleep-disordered breathing, such as sleep apnea, disrupts oxygen delivery, leading to fragmented sleep and daytime fatigue. Sleep apnea, a condition in which breathing repeatedly stops and starts during sleep, can have serious consequences for oxygen transport. In obstructive sleep apnea, the airway collapses, blocking airflow despite continued efforts to breathe. Oxygen levels drop, and carbon dioxide accumulates, triggering a brief arousal that restores breathing. These cycles can occur hundreds of times a night, preventing restorative sleep and straining the cardiovascular system. Over time, chronic intermittent hypoxia, the repeated drops in oxygen, can lead to high blood pressure, heart disease, and other complications. Treatment, often involving a device that keeps the airway open with positive pressure, restores normal breathing and oxygen delivery, improving both sleep quality and health. Aging affects oxygen transport in subtle but important ways. Lung capacity gradually declines, and the alveoli lose some of their elasticity, reducing the efficiency of gas exchange. The heart's pumping capacity may decrease, and blood vessels stiffen, increasing resistance to blood flow. Red blood cell production slows, and the bone marrow's responsiveness to erythropoietin diminishes. These changes are gradual and vary widely among individuals, influenced by genetics, lifestyle, and overall health. Regular physical activity, a balanced diet, and avoidance of smoking can slow age-related declines, preserving oxygen transport capacity and supporting vitality into later life. The study of oxygen transport has a rich history, intertwined with the development of physiology and medicine. In the 17th century, scientists began to understand that air contains a component essential for life, though they did not yet know it was oxygen. In the 18th century, oxygen was isolated and identified by several researchers, including Joseph Priestley and Carl Wilhelm Scheele. The 19th century brought discoveries about hemoglobin and the role of red blood cells. In the 20th century, researchers elucidated the molecular structure of hemoglobin, the mechanisms of oxygen binding, and the regulation of red blood cell production. Each generation of scientists built on the work of those before, gradually revealing the intricate beauty of oxygen transport. Christian Bohr, the Danish physiologist after whom the Bohr effect is named, made his discovery in the early 20th century. He found that carbon dioxide and acidity shift the oxygen-hemoglobin dissociation curve, enhancing oxygen release in tissues. His son, Niels Bohr, would go on to win the Nobel Prize in Physics for his work on atomic structure, a reminder that scientific inquiry often runs in families, with curiosity and rigor passed from one generation to the next. The Bohr effect remains a cornerstone of our understanding of oxygen transport, a principle taught to every student of physiology and medicine. Max Perutz, a molecular biologist, determined the three-dimensional structure of hemoglobin in the 1960s, using X-ray crystallography. His work revealed how the four subunits of hemoglobin fit together and how oxygen binding causes subtle shifts in their arrangement. This structural insight explained cooperativity, the phenomenon by which binding of one oxygen molecule makes it easier for the next to bind. Perutz' achievement earned him the Nobel Prize in Chemistry and opened the door to understanding other proteins at the molecular level. His legacy is a testament to the power of combining chemistry, physics, and biology to unravel the mechanisms of life. The story of oxygen transport is also a story of evolution, adaptation, and cooperation. From the ancient symbiosis that gave rise to mitochondria, to the development of hemoglobin in early vertebrates, to the fine-tuning of regulatory mechanisms in mammals, each step reflects the interplay of chance and necessity. Oxygen transport is not a single invention, but a layered system, with multiple components that work together seamlessly. It is a system that has been tested and refined over hundreds of millions of years, shaped by the challenges of diverse environments and the demands of active, complex life. And so, as you rest here, breathing softly, your body continues its quiet work. Oxygen flows from air to blood, from blood to cells, supporting the myriad reactions that sustain you. Carbon dioxide travels in reverse, expelled with each breath. Hemoglobin shifts between its tense and relaxed forms. Red blood cells flex and squeeze through capillaries, and the heart beats on, steady and strong. These processes, though intricate, unfold with a simplicity born of deep evolutionary wisdom. You need not think about them, need not direct them. They simply happen, a gift of your biology, a legacy of life's long history on this oxygen-rich planet. The story of oxygen transport carries us into the realm of medical understanding, where centuries of careful observation have revealed how fragile and resilient this system can be. Anemia, a condition where the blood carries insufficient oxygen-carrying capacity, comes in many quiet forms. Iron deficiency anemia, the most common type worldwide, develops when the body lacks enough iron to build hemoglobin molecules. Without adequate iron, red blood cells emerge smaller and paler than usual, each one carrying less hemoglobin, each breath delivering slightly less oxygen to waiting tissues. The fatigue that follows is the body's gentle signal that something in this ancient partnership needs attention. Treatment with iron supplementation allows the bone marrow to resume its full production, and over weeks, the blood regains its oxygen-carrying strength. This process reminds us that even robust systems sometimes need the simplest elements restored. Vitamin B12 and folate deficiencies create a different kind of anemia, one where red blood cells grow abnormally large but remain fewer in number. These vitamins play essential roles in DNA synthesis, and without them, the bone marrow struggles to complete cell division properly. The red blood cells that do emerge are oversized and fragile. Their membranes stretch thin, their lifespan shortened. This megaloblastic anemia develops slowly, often over months or years, as the body's stores of these vitamins gradually deplete. The nervous system, which also depends on vitamin B12, may show signs of distress before the blood changes become severe. Supplementation reverses the process, allowing normal cell division to resume, and the bone marrow once again produces properly sized, long-lived red blood cells. The body's forgiveness is remarkable, its capacity to heal when given what it needs. Hemolytic anemias occur when red blood cells break apart prematurely, their membranes rupturing before their natural lifespan ends. This can happen for many reasons. Autoimmune conditions where the body mistakenly attacks its own cells, genetic disorders affecting membrane proteins, infections that damage cell structures, or exposure to certain medications. When red blood cells hemolyze, they release their hemoglobin into the plasma, where it must be quickly processed to prevent damage to the kidneys. The spleen and liver work overtime to clear the debris, and the bone marrow accelerates production to replace lost cells. The result is a delicate balance, the body running faster just to stay in place. In some cases, The bone marrow can compensate fully, maintaining adequate oxygen delivery despite the ongoing destruction. In others, medical intervention helps slow the hemolysis or support the marrow's efforts. Sickle cell disease, which we touched upon earlier, presents unique challenges in oxygen transport. The abnormal hemoglobin causes red blood cells to deform under low oxygen conditions, but the consequences extend beyond simple shape changes. These sickled cells become sticky, adhering to blood vessel walls and to each other, creating blockages in small capillaries. These vaso-occlusive crises cause sudden, severe pain as tissues downstream from the blockage become starved of oxygen. Over time, repeated crises damage organs, particularly the spleen, kidneys, and bones. Yet people with sickle cell disease develop remarkable resilience, learning to manage their condition, avoid triggers, and seek treatment promptly when crises occur. Modern therapies, including hydroxyurea, which increases fetal hemoglobin production, have transformed outcomes, allowing the body's own developmental programs to provide relief. Thalassemias represent another category of genetic hemoglobin disorders, ones where the production of globin chains becomes imbalanced. In beta thalassemia, the beta chains are underproduced, leaving excess alpha chains that precipitate inside red blood cells, damaging them from within. In alpha thalassemia, the reverse occurs. The severity depends on how many genes are affected, ranging from silent carriers with no symptoms to severe forms requiring regular blood transfusions. The bone marrow responds to the chronic anemia by expanding dramatically, sometimes causing changes in bone structure. The body tries valiantly to compensate, but the fundamental imbalance in protein production creates red blood cells that cannot survive their normal lifespan. Regular transfusions provide functional red blood cells, while chelation therapy removes the excess iron that accumulates from transfused blood. The management of thalassemia shows medicine working in partnership with the body's own efforts. Polycythemia, the opposite condition where too many red blood cells are produced, also disrupts oxygen transport, though in a different way. When the blood becomes too thick with red blood cells, it flows more slowly through vessels, increasing the risk of clots. Primary polycythemia, also called polycythemia vera, arises from a mutation in bone marrow cells that causes uncontrolled red blood cell production. Secondary polycythemia develops as a response to chronic low oxygen levels, the body reasonably but excessively trying to improve oxygen delivery. People living at very high altitudes, those with chronic lung disease, or individuals with certain tumors that produce erythropoietin may develop this condition. Treatment often involves phlebotomy, the regular removal of blood, bringing the red blood cell count back toward normal and allowing blood to flow more easily. Even in excess, the system can be gently rebalanced. Carbon monoxide poisoning reveals the vulnerability of hemoglobin's binding sites with particular clarity. Carbon monoxide, produced by incomplete combustion, binds to hemoglobin with an affinity about 200 times greater than oxygen. When carbon monoxide occupies hemoglobin's binding sites, oxygen cannot attach, and the blood's carrying capacity plummets. Even more insidiously, carbon monoxide binding changes hemoglobin's shape in a way that makes it hold onto any remaining oxygen more tightly, preventing oxygen release to tissues. The result is severe tissue hypoxia despite normal breathing efforts. The treatment is elegant in its simplicity, high concentrations of oxygen, sometimes delivered under pressure in hyperbaric chambers. The oxygen gradually displaces carbon monoxide from hemoglobin's embrace, though the process takes time because carbon monoxide departs slowly. The body eventually clears all the carbon monoxide. Hemoglobin returns to normal function, and oxygen transport resumes its usual patterns. Methemoglobinemia, which we mentioned earlier, can also occur as an acquired condition, not just a rare genetic one. Certain medications, particularly some local anesthetics and antibiotics, can oxidize hemoglobin's iron, converting it from the ferrous to ferric state. When methemoglobin levels rise above about 10% of total hemoglobin, the blood takes on a characteristic chocolate brown color, and oxygen delivery begins to decline noticeably. At higher levels, symptoms of hypoxia appear: shortness of breath, confusion, headache. The treatment, methylene blue, acts as an electron donor, helping to reduce the iron back to its functional ferrous state. The body also has its own methemoglobin reductase, enzymes working constantly, but they can be overwhelmed by rapid oxidation. Once the oxidizing agent is removed and reduction therapy given, normal hemoglobin function returns, another example of the body's chemical resilience. The placenta, that remarkable temporary organ, creates a specialized environment for oxygen transfer between maternal and fetal blood. The two blood supplies never actually mix. Instead, they flow past each other separated by thin membranes and structures called chorionic villi. Oxygen diffuses from maternal blood, where it's more abundant, into fetal blood, where it's less concentrated. The fetal hemoglobin's higher oxygen affinity, which we discussed earlier, facilitates this transfer, pulling oxygen across the gradient. Carbon dioxide moves in the opposite direction, from fetal to maternal blood, to be exhaled by the mother's lungs. The placenta also produces hormones that increase the mother's breathing rate and red blood cell production, ensuring adequate oxygen supply for two. This system works continuously throughout pregnancy. Every breath the mother takes shared in a sense with the developing child. At birth, the newborn's first breaths fill the lungs with air, and the entire circulatory system reorganizes within minutes, closing fetal shunts and redirecting blood through the now functional lungs. Premature infants sometimes face challenges with oxygen transport because their systems aren't fully mature. The lungs may lack sufficient surfactant, making breathing difficult. Red blood cell production may be sluggish, and erythropoietin levels may not respond appropriately to oxygen needs. Fetal hemoglobin, while excellent for intrauterine life, releases oxygen less readily than adult hemoglobin, which can be a disadvantage when the infant needs to extract oxygen from their own lungs. Modern neonatal care supports these immature systems. Supplemental oxygen when needed, surfactant replacement therapy, sometimes erythropoietin injections to stimulate red blood cell production. Most premature infants' oxygen transport systems mature over weeks to months, gradually taking over the functions that medical support initially provided. The body's developmental program continues outside the womb, eventually reaching the same endpoints, just on a delayed schedule. High-altitude mountaineering pushes oxygen transport to its absolute limits. On the summit of Mount Everest, the atmosphere pressure is only about 1/3 that at sea level, meaning each breath contains far fewer oxygen molecules. Even with the adaptations we discussed earlier, increased red blood cell production, improved capillary density, enhanced oxygen extraction, the body operates at the edge of viability. Climbers at extreme altitude breathe much faster than normal, their respiratory rate sometimes doubling or tripling. Their hearts beat rapidly, trying to circulate the oxygen-poor blood more quickly. Despite these efforts, arterial oxygen saturation may drop to 60 or 70%, levels that would cause unconsciousness at sea level. The body tolerates this only because the adaptations work together synergistically. Climbers also typically use supplemental oxygen above certain altitudes, directly increasing the oxygen content of each breath. Descending to lower altitudes brings rapid relief as oxygen becomes more abundant with each step downward. Deep-sea diving creates opposite challenges for oxygen transport. Under pressure, gases dissolve more readily in blood and tissues, following Henry's law. Divers breathing compressed air absorb more nitrogen into their bloodstream and body tissues. This isn't immediately problematic, but if they ascend too quickly, the dissolved nitrogen comes out of solution as bubbles, like opening a carbonated beverage. These bubbles can block blood vessels, causing decompression sickness, also known as the bends. Oxygen itself becomes toxic at high partial pressures, which is why deep divers often use special gas mixtures with reduced oxygen percentages. The body's oxygen transport system, evolved for atmospheric pressure, must be carefully managed in these extreme environments. Controlled ascent rates allow dissolved gases to be eliminated gradually through the lungs, preventing bubble formation. The body's resilience extends even here, but only when we respect the physical laws governing gas behavior. Breath hold diving, practiced by free divers and traditionally by pearl divers and AMA divers of Japan and Korea, relies entirely on the oxygen stored in the body at the moment of submersion. This stored oxygen exists in three main pools: air in the lungs, oxygen bound to hemoglobin in the blood, and oxygen bound to myoglobin in muscles. Trained freedivers can hold their breath for many minutes, descending to remarkable depths. During the dive, the body activates the mammalian diving reflex. Heart rate slows dramatically, blood vessels in the limbs constrict, and blood flow concentrates on the brain and heart. This conserves oxygen for the most critical organs. Hemoglobin gradually releases its oxygen to tissues, saturation levels dropping steadily. The spleen contracts, releasing stored red blood cells into circulation, providing a small but meaningful boost to oxygen carrying capacity. As the diver ascends, the decreasing pressure causes oxygen levels to drop further, and the final meters before surfacing are the most dangerous, when shallow water blackout can occur if oxygen falls too low. Successful free divers develop an intimate awareness of their body's oxygen status, learning to read subtle signals and surface before reaching critical limits. The respiratory control centers in the brainstem monitor carbon dioxide levels more closely than oxygen levels under normal circumstances. This might seem counterintuitive, but it reflects an evolutionary reality. In natural environments, if carbon dioxide is rising, oxygen is almost certainly falling, so monitoring CO2 provides an effective proxy. However, in some situations, this can create problems. People with chronic obstructive pulmonary disease, COPD, sometimes retain carbon dioxide chronically, and their respiratory control centers adapt to these higher levels, becoming less sensitive to CO2. In these individuals, low oxygen becomes the primary drive to breathe. If they're given too much supplemental oxygen, eliminating the hypoxic drive, their breathing rate may slow dangerously. This is why oxygen therapy in COPD must be carefully titrated, providing enough oxygen to prevent tissue damage, but not so much that it suppresses the drive to breathe. The body's control systems, usually so reliable, can be caught in difficult situations when disease alters the normal relationships between gases. Sleep apnea creates cyclical disruptions in oxygen transport. During apneic episodes, breathing stops for seconds to minutes, and oxygen saturation gradually falls while carbon dioxide rises. Eventually, the oxygen drop or CO2 rise triggers arousal, breathing resumes, and oxygen levels recover. This cycle can repeat dozens or even hundreds of times per night. The repeated oxygen desaturations stress the cardiovascular system, triggering sympathetic nervous system activation, raising blood pressure and heart rate. Over time, chronic intermittent hypoxia contributes to hypertension, heart disease, and stroke risk. The body tries to adapt, sometimes increasing red blood cell production in response to the repeated low oxygen episodes. But this is at best a partial compensation. Treatment with continuous positive airway pressure, CPAP, keeps airways open during sleep, preventing the apneas and allowing normal, stable oxygen levels throughout the night. The cardiovascular system can then rest and recover, no longer subjected to the repeated stress of oxygen desaturation and reoxygenation. Exercise training creates beneficial adaptations throughout the oxygen transport system, changes that develop gradually over weeks and months. The heart muscle strengthens, allowing it to pump more blood with each beat, increasing stroke volume. The total number of red blood cells increases slightly, expanding oxygen carrying capacity. Capillary density in muscles improves, shortening the diffusion distance between blood and muscle fibers. Mitochondria within muscle cells become more numerous and efficient, better able to use delivered oxygen. The result is improved endurance. The same exercise intensity requires less effort, feels easier, and can be sustained longer. Elite endurance athletes develop these adaptations to extraordinary degrees, their oxygen transport and utilization systems operating at the upper limits of human capability. Yet the same basic adaptations occur in anyone who exercises regularly, the body responding to the repeated challenge by becoming more efficient at delivering and using oxygen. The aging process gradually affects oxygen transport, though healthy aging individuals maintain adequate function for normal activities. Red blood cell production slows somewhat, and the bone marrow's reserve capacity diminishes. Hemoglobin levels may decline slightly, especially in very elderly individuals. The cardiovascular system becomes less responsive, maximum heart rate decreasing with age. Lung function declines, with reduced elasticity and decreased surface area for gas exchange. Mitochondria become less efficient, and muscle mass decreases, reducing overall oxygen utilization capacity. These changes are gradual and vary greatly between individuals, influenced by genetics, lifestyle, and overall health. Regular physical activity throughout life helps maintain oxygen transport function, preserving capillary density, mitochondrial function, and cardiovascular fitness. The body systems, while aging, retain remarkable function when consistently used and maintained. Smoking damages oxygen transport through multiple mechanisms, all of them detrimental. Carbon monoxide in smoke binds to hemoglobin, chronically reducing oxygen carrying capacity. Nicotine constricts blood vessels, reducing blood flow to tissues. Chronic smoke exposure damages lung tissue, reducing the surface area for gas exchange and impairing oxygen uptake. The body responds to chronic mild hypoxia by increasing red blood cell production, which might seem beneficial, but the resulting polycythemia thickens the blood, slowing flow and increasing clot risk. Smoking also damages blood vessel walls, promoting atherosclerosis and further impairing circulation. The cumulative effect is a compromised oxygen transport system, less able to meet the body's needs. The encouraging reality is that smoking cessation allows many of these changes to reverse. Carbon monoxide clears from the blood within hours. Circulation begins to improve within weeks. Lung function gradually recovers over months to years. Red blood cell counts normalize. The body's capacity for healing, given the chance, is profound. Blood donation temporarily reduces oxygen carrying capacity, but the healthy body compensates easily. A typical donation removes about 10% of total blood volume, including the red blood cells it contains. Plasma volume recovers within 24 to 48 hours as the body retains fluid and adjusts blood volume. Red blood cell replacement takes longer, about 4 to 8 weeks, as the bone marrow produces new cells to replace those donated. During this recovery period, hemoglobin levels are slightly lower than normal, and oxygen carrying capacity is modestly reduced. Most donors experience no symptoms, their bodies compensating through slightly increased heart rate and cardiac output. The bone marrow's ability to ramp up production, increasing red blood cell output by several-fold when needed, ensures recovery. Iron stores may be depleted by repeated donations, which is why donors are often advised to take iron supplements and why donation frequency is limited. The system's resilience allows this act of generosity without lasting impact on the donor's health. Blood transfusion provides immediate restoration of oxygen-carrying capacity in situations where the body cannot compensate on its own. Severe bleeding, whether from trauma, surgery, or medical conditions, can reduce red blood cell mass so rapidly that the bone marrow cannot keep pace. Transfusing packed red blood cells, which are donated red blood cells with most plasma removed, directly replaces lost oxygen-carrying capacity. The transfused cells function immediately, binding and transporting oxygen just as the recipient's own cells do. These transfused cells have a somewhat shorter lifespan than fresh cells, but they still circulate for weeks, providing a bridge until the recipient's bone marrow can restore normal red blood cell levels. Blood typing and cross-matching ensure compatibility, preventing immune reactions against the transfused cells. Modern blood banking, with careful screening and testing, has made transfusion remarkably safe, a medical intervention that saves countless lives by supporting the body's oxygen transport when it's most compromised. Artificial oxygen carriers have been researched for decades as potential alternatives to blood transfusion. Hemoglobin-based oxygen carriers use purified hemoglobin, sometimes chemically modified or encapsulated, to transport oxygen without requiring intact red blood cells. Perfluorocarbon emulsions dissolve oxygen directly, carrying it physically rather than through chemical binding. These products could potentially be stored longer than donated blood, wouldn't require blood type matching, and could be manufactured in large quantities. However, developing safe and effective artificial oxygen carriers has proven challenging. Free hemoglobin outside red blood cells can cause vasoconstriction and kidney damage. Perfluorocarbons carry less oxygen than blood and require high inspired oxygen concentrations. None have yet achieved widespread clinical use, though research continues. The natural system, with its red blood cells, hemoglobin carefully contained and regulated, remains more sophisticated than our attempts to replicate it. Hyperbaric oxygen therapy uses elevated atmospheric pressure to increase the amount of oxygen dissolved directly in plasma. At normal atmospheric pressure, dissolved oxygen contributes little to total oxygen content, as we discussed earlier. But at 2 or 3 times atmospheric pressure, the amount of dissolved oxygen increases proportionally, becoming physiologically significant. This can be beneficial in certain medical conditions: carbon monoxide poisoning, where hemoglobin is blocked, severe infections with certain bacteria that are killed by high oxygen levels, wound healing in tissues with poor blood flow. The patient breathes pure oxygen in a pressurized chamber, and the dissolved oxygen reaches tissues even where red blood cell delivery is compromised. Treatments typically last 1 to 2 hours and may be repeated daily for weeks. The therapy has limitations and risks, oxygen toxicity being the main concern. But in selected situations, it harnesses physical principles to temporarily bypass some of the normal oxygen transport system. Gene therapy for hemoglobin disorders represents a frontier in treating oxygen transport diseases. The approach involves extracting a patient's own bone marrow stem cells, inserting a functional copy of the defective gene using viral vectors, and then returning the corrected cells to the patient. These genetically modified stem cells then produce healthy hemoglobin, either beta globin in beta thalassemia or corrected hemoglobin in sickle cell disease. Early clinical trials have shown remarkable success, with some patients becoming transfusion independent or experiencing dramatic reductions in sickle cell crises. The therapy essentially cures the disease at its source, correcting the genetic instruction that has been causing problems since birth. The modified cells are the patient's own, so there's no rejection risk. This approach shows medicine working at the most fundamental level, editing the code that determines hemoglobin structure, allowing the body's own production systems to then create functional oxygen transport molecules. The study of oxygen transport has deep historical roots, though early investigators didn't fully understand what they were observing. In the 17th century, Robert Hooke and Robert Boyle demonstrated that animals needed air to survive, though they didn't yet know about oxygen. In 1774, Joseph Priestley discovered oxygen, which he called dephlogisticated air, and noted that mice lived longer in it. Antoine Lavoisier recognized oxygen as an element and understood that respiration was a form of combustion, though he thought it occurred in the lungs. In the 19th century, scientists discovered that blood carries oxygen and that hemoglobin is responsible. The precise structure of hemoglobin wasn't determined until the mid-20th century, when Max Perutz used X-ray crystallography to reveal its molecular architecture, work that earned him a Nobel Prize. Each generation of scientists built on previous discoveries, gradually illuminating the elegant mechanisms we've explored. The knowledge that now seems so clear was painstakingly assembled over centuries of careful observation and experiment. The oxygen transport system connects us to the atmosphere in a continuous exchange that has been ongoing since our first breath. Every inhalation brings molecules that may have been exhaled by someone across the world, by trees in distant forests, by ocean phytoplankton. The oxygen we breathe today was produced by photosynthetic organisms over millions of years, split from water molecules using energy from sunlight. Our hemoglobin, carrying iron forged in ancient stars, binds these oxygen molecules and delivers them to our cells. The carbon dioxide we return to the atmosphere enters the carbon cycle, eventually taken up by plants to build new organic molecules. We are participants in planetary cycles, our breath connecting us to all life, past and present. This perspective, seeing oxygen transport not just as personal physiology, but as part of Earth's great biogeochemical cycles, reveals our deep integration with the living world. The regulation of oxygen transport involves constant communication between tissues, blood, and control centers. Sensors in blood vessels, particularly the carotid bodies and aortic bodies, continuously monitor oxygen levels. These chemoreceptors send signals to the brainstem, which adjusts breathing rate and depth. The kidneys sense oxygen delivery and modulate erythropoietin production, controlling red blood cell production over days and weeks. Local tissues regulate their own blood flow through metabolic signals, ensuring that active areas receive more blood. The heart responds to demands for increased cardiac output, pumping faster and more forcefully during exercise or stress. All these regulatory loops operate simultaneously, creating a coordinated response that maintains oxygen delivery across varying conditions. The system is self-adjusting, requiring no conscious oversight, operating reliably throughout our lives. This autonomic regulation frees our conscious minds from the burden of managing our own physiology, allowing us to focus on other concerns while our bodies quietly maintain themselves. Temperature affects oxygen transport in ways that extend beyond hemoglobin's oxygen affinity. Cold temperatures increase blood viscosity, making it flow more sluggishly through vessels. Peripheral vasoconstriction in response to cold reduces blood flow to skin and extremities, conserving heat but also reducing oxygen delivery to these areas. This is why fingers and toes are most vulnerable to frostbite. Reduced blood flow means reduced oxygen and nutrient delivery, making tissues more susceptible to damage. Conversely, heat causes vasodilation and decreased blood viscosity, increasing blood flow to the skin for cooling. The body prioritizes temperature regulation, sometimes at the expense of optimal oxygen delivery to non-essential tissues. Extreme cold can cause hemoglobin to hold oxygen more tightly. As we discussed, while extreme heat facilitates oxygen release, but may compromise cardiovascular function through excessive demands on the heart. The oxygen transport system must operate across a range of temperatures, adapting its function to thermal conditions while maintaining adequate delivery to vital organs. The spleen's role in oxygen transport extends beyond quality control of red blood cells. The spleen also serves as a reservoir, storing red blood cells that can be released when needed. During exercise, stress, or situations requiring increased oxygen carrying capacity, the spleen contracts, squeezing stored red blood cells into circulation. This can increase the hematocrit by several percentage points, meaningfully boosting oxygen transport capacity. Horses have particularly large spleens relative to body size and can increase their red blood cell count by 50% or more through splenic contraction, an adaptation for their evolution as prey animals needing sudden bursts of speed. Humans have smaller spleens and more modest splenic reserves, but the mechanism still contributes to our ability to respond to acute demands. People who have had their spleens removed due to injury or disease lose this reserve capacity. Though they generally adapt well, their bodies compensating through other mechanisms. Erythrocytosis, the condition of having elevated red blood cell counts, can be primary or secondary. Primary erythrocytosis arises from mutations in bone marrow cells that cause excessive production. Secondary erythrocytosis develops in response to stimuli, most commonly chronic hypoxia. People with cyanotic heart disease, where oxygen-poor and oxygen-rich blood mix abnormally, often develop secondary erythrocytosis as their bodies try to compensate for chronically low oxygen saturation. Similarly, individuals with chronic lung diseases may have elevated red blood cell counts. Interestingly, some elite endurance athletes have naturally high red blood cell counts, possibly due to genetic variations affecting erythropoietin signaling or red blood cell production. This raises complex questions in sports about distinguishing natural variation from artificial enhancement. The body's response to low oxygen, increasing red blood cell production, is fundamentally adaptive, but like many physiological responses, it can become maladaptive when excessive or when the underlying problem cannot be corrected. The discovery of blood types by Carl Landsteiner in 1901 revolutionized medicine and our understanding of blood. He identified the A, B, and O blood groups, recognizing that red blood cells carry different antigens on their surfaces and that plasma contains antibodies against foreign antigens. This explained why some blood transfusions succeeded while others caused fatal reactions. The ABO system relates to oxygen transport indirectly. The antigens don't affect hemoglobin function, but they're crucial for transfusion safety. Later, the RH system was discovered, adding another layer of complexity. Blood typing ensures that transfused red blood cells won't be attacked by the recipient's immune system, allowing these cells to circulate normally and transport oxygen. The existence of different blood types is a reminder that genetic variation affects even fundamental systems like red blood cells, creating diversity that has implications for medicine and evolutionary history. Genetic variations in hemoglobin are remarkably common in human populations. Beyond the major variants like sickle cell and thalassemias, hundreds of hemoglobin variants have been identified, most rare and many without clinical significance. Some alter oxygen affinity slightly, causing mild polycythemia or mild anemia, but no serious symptoms. Others affect hemoglobin stability or solubility. The high frequency of hemoglobin variants in certain populations reflects evolutionary pressures, particularly for malaria. Variants that confer some protection against malaria have been maintained in populations where malaria is endemic, despite causing disease in individuals who inherit two copies. This is balanced polymorphism, where genetic diversity is maintained because different variants have advantages in different circumstances. Our hemoglobin, that crucial oxygen transport molecule, carries the signatures of our evolutionary history, adapted and shaped by the diseases and environments our ancestors encountered. The oxygen cascade, the series of steps by which oxygen moves from atmosphere to mitochondria, reveals the elegant efficiency of the transport system. Oxygen partial pressure is highest in the atmosphere, around 160 millimeters of mercury at sea level. It drops slightly in the airways due to humidification and mixing with residual air. In the alveoli, it's about 100 millimeters of mercury. Diffusing into arterial blood, it remains near 100 millimeters of mercury. In systemic capillaries, it drops to around 40 millimeters of mercury as oxygen diffuses into tissues. Inside cells, oxygen partial pressure is lower still, perhaps 20 millimeters of mercury or less. At the mitochondria, where oxygen is consumed, the partial pressure approaches zero. This cascade of decreasing pressures drives oxygen movement at each step from high to low, without requiring energy expenditure for the diffusion itself. The system harnesses the natural tendency of gases to move down concentration gradients, using passive physical processes to accomplish sophisticated transport. Energy is spent pumping blood and breathing air, but the actual transfer of oxygen across membranes happens spontaneously, driven by pressure differences. The relationship between oxygen and carbon dioxide transport is intimately connected through hemoglobin. We've discussed the Bohr effect, where CO2 and hydrogen ions reduce hemoglobin's oxygen affinity. There's also a reciprocal effect. Deoxygenated hemoglobin binds CO2 and hydrogen ions more readily than oxygenated hemoglobin. This is called the Haldane effect, and it enhances CO2 transport from tissues to lungs. In tissues, as hemoglobin releases oxygen, it simultaneously becomes better at picking up CO2 and buffering hydrogen ions. In the lungs, as hemoglobin binds oxygen, it releases CO2 and hydrogen ions, facilitating CO2 elimination. These reciprocal effects mean that oxygen and carbon dioxide transport are not independent processes, but are mechanistically linked through hemoglobin's changing properties. The molecule that carries oxygen also participates in removing the waste product of oxygen consumption, an elegant economy of function. Breathing patterns affect oxygen transport in subtle ways. Slow, deep breathing maximizes alveolar ventilation and gas exchange efficiency. Rapid, shallow breathing may move the same total volume of air, but ventilates the airways more than the alveoli, reducing efficiency. Breath holding allows oxygen extraction from the lungs to continue even without fresh air entering, which is why breath hold diverse exhale partially before diving, reducing lung volume, but keeping some oxygen available for gradual absorption. Hyperventilation before breath holding, once common among divers, is now recognized as dangerous because it lowers CO2 levels without significantly increasing oxygen stores, delaying the urge to breathe and increasing the risk of losing consciousness underwater. These patterns show how breathing mechanics interact with oxygen transport, the rhythm and depth of breathing affecting how efficiently oxygen enters the blood. The psychological state can influence oxygen transport through effects on breathing and cardiovascular function. Anxiety often causes rapid breathing, sometimes leading to hyperventilation, which lowers CO2 levels and can cause symptoms like dizziness and tingling. Stress activates the sympathetic nervous system, increasing heart rate and blood pressure, redirecting blood flow to muscles in the fight or flight response. Relaxation and meditation, slow breathing and heart rate, promoting efficient gas exchange and reducing cardiovascular work. The mind-body connection extends to oxygen transport, our thoughts and emotions influencing the physiological processes that sustain us. Practices that promote calm, slow breathing, mindfulness, relaxation, support optimal oxygen transport function. allowing the system to operate efficiently without the stress-induced demands that increase oxygen consumption while potentially compromising delivery. Oxygen transport continues even during sleep, the body's needs persisting through rest. During sleep, metabolic rate decreases and oxygen consumption drops by about 10 to 15%. Breathing slows and becomes more regular during deep sleep, though it may become irregular during REM sleep. Heart rate decreases and blood pressure drops. The body uses this reduced demand period for repair and restoration, but oxygen delivery must continue uninterrupted. The respiratory control centers remain active, adjusting breathing to maintain appropriate oxygen and CO2 levels. The heart continues its rhythmic pumping, circulating blood through sleeping tissues. Red blood cells continue their endless circuits, binding and releasing oxygen, carrying out their function whether we're awake or asleep. The autonomic nature of oxygen transport allows it to continue without conscious awareness, supporting life through all states of consciousness. As we near the end of our exploration, we can reflect on the profound reliability of oxygen transport. From the first breath at birth to the last breath at life's end, this system operates continuously, adapting to changing demands, compensating for challenges, maintaining the oxygen supply that every cell requires. It operates without our conscious control or awareness, a gift of our evolutionary heritage and our developmental biology. The hemoglobin molecules in our blood are carrying out the same function they've performed for hundreds of millions of years, a molecular solution to oxygen transport that has been refined and optimized across vast spans of time. The red blood cells coursing through our vessels are the descendants of an unbroken lineage stretching back to our first moments, each generation replaced by the next in an ongoing renewal. The breath we draw connects us to the atmosphere, to the photosynthetic organisms that produce oxygen, to the great cycles of life on Earth. Understanding oxygen transport enriches our appreciation of our own biology. We are not simple machines, but complex, self-regulating systems with remarkable capacities for adaptation and compensation. The oxygen transport system exemplifies biological elegance. Multiple components working in coordination, regulated by feedback loops, responsive to changing conditions, resilient in the face of challenges. It reminds us that our bodies are worthy of respect and care, that the systems sustaining us are both robust and, in some ways, delicate. Supporting our oxygen transport system through healthy lifestyle choices, not smoking, staying active, maintaining good nutrition, managing chronic conditions, allows it to function optimally, supporting our health and vitality throughout our lives. The science of oxygen transport continues to advance. Researchers are developing better treatments for hemoglobin disorders, more sophisticated understanding of high-altitude adaptation, improved artificial oxygen carriers, and novel therapies for conditions affecting oxygen delivery. Each discovery adds to our knowledge, revealing new details of mechanisms we thought we understood, finding connections we hadn't recognized. The story of oxygen transport is not complete, but ongoing. Each generation of scientists adding their observations and insights to the accumulated understanding. We are privileged to live in an era when so much is known, when the molecular details of hemoglobin function have been revealed, when genetic therapies can correct the very instructions that determine hemoglobin structure. Yet for all our scientific understanding, there remains something almost miraculous about oxygen transport. That a molecule like hemoglobin can bind oxygen loosely enough to release it where needed, but tightly enough to carry it through the bloodstream. That this binding and release can be regulated by pH and CO2 and temperature. That millions of red blood cells can circulate simultaneously, each carrying millions of hemoglobin molecules, all working in coordination to supply trillions of cells with oxygen. The complexity and reliability of this system inspire wonder. We are, in every moment, sustained by processes of exquisite sophistication, processes that require no conscious effort, that simply work day after day, year after year. As you rest now, your oxygen transport system continues its quiet work. Your lungs expand and contract, drawing in oxygen-rich air. Oxygen diffuses across alveolar membranes into your blood. Hemoglobin molecules bind oxygen, their shape shifting subtly as iron atoms embrace oxygen molecules. Your heart pumps steadily, sending oxygenated blood through arteries to capillaries throughout your body. In those tiny vessels, oxygen releases from hemoglobin and diffuses into cells. Mitochondria consume that oxygen, producing the energy that powers cellular processes. Carbon dioxide, the waste product, diffuses back into blood, is carried to your lungs, and is exhaled. This cycle repeats with every breath, every heartbeat, sustaining you as you read these words, as you drift towards sleep throughout the night ahead. The knowledge we've explored tonight is meant not to burden you, but to offer appreciation for the remarkable biology that sustains you. You need do nothing to maintain oxygen transport. It maintains itself, regulated by systems perfected over evolutionary time, operating reliably without your conscious oversight. You can rest easy, knowing that your body continues its essential work, that oxygen will be delivered to every cell, that the processes supporting life will continue through the night. This is the gift of biological complexity. Systems so sophisticated and well-regulated that they free us from the need to consciously manage our own survival, allowing us to rest, to sleep, to dream, while our bodies quietly take care of themselves. So as you settle into rest, let go of any effort, any tension. Your oxygen transport system doesn't need your help or attention. It will continue its work through the night, as it has every night of your life. The red blood cells circulating through your vessels right now will complete thousands of circuits before morning, each one carrying its cargo of oxygen, each one contributing to your vitality. The hemoglobin molecules in those cells will bind and release oxygen countless times, their function unchanged by whether you're awake or asleep, active or at rest. Your breathing will slow and deepen as you relax, becoming more efficient, more peaceful. Your heart rate will settle into its resting rhythm, pumping steadily, reliably. There is comfort in understanding the systems that sustain us, in knowing that we are supported by biology that is both ancient and remarkably effective. The oxygen transport mechanisms we've explored tonight connect you to all other humans, to our evolutionary ancestors, to the great web of aerobic life on Earth. Every breath you take participates in planetary cycles. Every oxygen molecule, your hemoglobin, carries links you to the photosynthetic organisms that produced it. You are part of something vast and interconnected, sustained by processes that have supported life for hundreds of millions of years and will continue long into the future. Rest now, knowing that you're cared for by your own biology, that the systems sustaining you are reliable and resilient, that oxygen will continue to flow to every cell throughout the night. Let the knowledge we've explored settle gently in your mind, not as facts to be memorized, but as appreciation for the elegant complexity of life. You are a marvel of biological engineering, sustained by processes of extraordinary sophistication. Yet you need do nothing but rest. Your body knows what to do, has always known, will continue to know. Trust in that ancient wisdom, in the systems perfected over deep time, in the quiet reliability of oxygen transport. Sleep well, breathe easily, and let your oxygen transport system